Bismillahirrahmanirrahim. Assalamu alaikum, ladies. Bismillahirrahmanirrahim. Assalamu alaikum, ladies and gentlemen. I'm Hazima Mozam, and I welcome you to the second day of online meeting on recent developments in drug discovery and medicinal chemistry. This event is organized jointly by Comstech in collaboration with HEJ Research Institute of Chemistry, International Center for Chemical and Biological Sciences. We will begin this session today with our first speaker, who is Dr. Hina Siddiqui. Dr. Hina Siddiqui is an associate professor at ICCBS, University of Karachi. The title of her talk today is Case Study, Successful Drug Discovery Made Through Medicinal Chemistry Approach. And with this, I would request His Excellency, Professor Dr. Iqbal Chaudhary, to kindly introduce and welcome our guest speaker today. Over to you, sir.
Bismillahirrahmanirrahim. Thank you, Hazima. What can be a greater pleasure than introducing someone you know since her childhood? Hina joined me when she was a baby. She's still very, very young and completed most of her studies under my supervision, then went forward to work in different laboratories of the world and establish herself as a very prominent young scientist. Hina got her PhD from HEJ Research Institute of Chemistry, International Center for Chemical and Biological Sciences, University of Karachi. During her doctoral studies, she went to Department of Chemistry, University of Kansas, USA, and got postdoctoral training at the Department of Chemistry, University of Tübingen in Germany. She has received a lot of external funding from different sources and published several very good papers in leading international journals. Her research is focused on multi-drug resistance reversal agents and drug repurposing and repositioning studies. Hina has supervised many young scientists and has over 3 dozen publication to her credit. She has also many patents. I requested Hina to focus more on workshop aspect of this event. I asked her to give examples of what medicinal chemistry can do. So presentation of Hina would largely be focused on how this entire concept of drug discovery and development has emerged and what are different strategies followed. Hina, over to you.
Thank you so much, sir. Such a great honor for me that you have introduced me. And whenever I present talk in front of you, my teacher, my heart rate goes up. So thank you so much, sir. So Hazima, can you see my slides?
Yes.
Yes, Dr. Hina.
Okay. So as sir mentioned that I'll be talking about in following 45 minutes or maybe 1 hour if Hazima allows me about the successful drug discoveries made through medicinal chemistry. I will mainly discuss examples from literature and some of the techniques and tools available in the medicinal chemistry which we can, our young students can utilize during their research while they are doing MedChem. And I'll briefly touch some of our work example in the medicinal chemistry. I want to start my presentation with a question, very, very simple question, and that how many molecules, organic molecules are possible on the face of the earth? Do we have answer to this question? Yes, of course we have. On approximation basis, approximately 500 proteins have been currently targeted in the proteomes, right? And it is estimated that the so-called drug-like chemical space is estimated about 10 power 63. That's huge. Small molecular structures. These are possible chemical entities starting from carbon 1 to carbon N, right? And so far, only 10 power 8 chemical entities, which we call new chemical entities or known chemical entities, have been synthesized, isolated from natural resources, semi-synthesized or biotransformed, or isolated from marine product, whatever. So it is still there is a huge need to synthesize more and more chemical space, more and more compounds, small molecules which are drug-like. And when I say drug-like, that means the compound should follow the Lipinski rule, right? And we all are aware of Lipinski rule because yesterday Saeed, Dr. Saeed has elaborated these stuff in really detail. So this is the high throughput screening paradigm, right? And so far we have 500 proteins targeted, and in order to target these proteins, we need to have some compounds to bind with these proteins so that we can come up some leads or some hits and followed by some leads to target these protein and eventually cure certain disease. So we need to, in order to do this, we need to interrogate the proteomes, we need to interrogate the genomes as well. So having said that, here is a picture. Picture A is basically representing the chemical space. Each dot present here is basically chemical entity, a chemical molecule, which I just mentioned that it is estimated that 10 power 63 drug-like molecule may exist, and you never know that one dot from this chemical space is a life-saving drug molecule, right? So our drug designing and development process is largely based on learning cycle, which is basically design, make, and test. This is what we medicinal chemists call learning cycle. It is used to nominate the molecules from chemical space, executing their synthesis and testing, and then analyzing the outcome, and eventually nominating the next round of the molecule. Only molecule that can synthesize will complete the learning cycle. Of course, we can design through in silico means and virtual screening we can do, of course, but eventually we need to have experimental models. We need to have these compounds actually in our hands so that we can test them, right? And once we have these molecules and we are testing them, we need to have, of course, we synthesize huge molecular libraries, but all the compound doesn't fit on a ideal profile of a drug. So there are certain parameters for a drug, for a compound to be a drug candidate. That include permeability. The compound has to pass through the blood-brain barriers, right? And triple B, what we call. So this is permeability, solubility, selectivity. It has to be selectively bind with the target which you are using for treatment of certain disease, and then it has to be metabolically stable, and certainly it need to have enough potency to treat certain disease, and of course synthetically accessible. What that means is basically the tools of synthetic chemistry which you are using are easy to handle in terms of whether in terms of chemicals, like you need to use accessible chemical which are readily available, should not be really expensive so that you cannot afford the synthesis, and then how can you afford the drug? So cheap, readily available starting materials, easily handle chemistry. So these are some parameters which we take into consideration while we talk about the MedChem, because in medicinal chemistry we need to synthesize large molecular libraries. And then certainly these are iteration which we need to consider. So having said that, the current slide represent a platform which we call, which we medicinal chemists call the library platform. So since our intention as a medicinal chemist are to probe chemical space with new molecular entities. So any type of research, medicinal chemistry or library platform always start with the idea. How we will get the idea is basically from bioactive lead molecules. You can think of some virtual screening, where you can do data mining, data searches, and of course you can also take your idea from any natural product which have certain biological activity, and then you can develop your molecular library based on that. So these are the bioactive lead or in silico screening, which gives you idea, and based on that idea you basically design molecules and design methodology. When we say methodology, that means it should be based on a minimal step, right? Synthetic step. So to the main scaffold of the drug library which you are synthesizing, or the molecular entities which you are synthesizing, should in medicinal chemistry we say 3 to 6 steps are enough, because we need to do multiple synthesis. We cannot have 50 step like total synthesis. We cannot perform it matchem while we are doing 50 step synthesis. So after that we produce libraries of the compounds, and in the modern ages there are multiple protocols are available, multiple technologies are available which can facilitate the synthesis. As far as synthesis is concerned, we can go for the flow chemistry, automated synthesizer, parallel synthesizer. So many technologies and equipments are available for the large number of library synthesis. And in addition to that, for the purification of the synthesized compound, there are also biotages are available which facilitate you for the automated purification of the compounds. So this is how you basically produce the library. I'm sorry, I just skipped. Okay. So this is how you basically produce the library, and after that comes the bioscreening. And this is the step where you basically develop multiple of bioassays, primary bioassays, in order to find out certain hits. Hits are those compounds which show some activity in vitro against certain targets. These targets can be enzymes, right? And some biochemical assays, cell-based assay maybe sometimes. So from this step, you of course get the hits, and then these hits become the idea for the next phase. Because after you have hit, you need to develop those compounds as a lead molecule. And when I say develop, that means lots of optimization, lots of functional group interconversion would take place in order to enhance the biological activities which you desire to have. Okay, as I discussed earlier, there is a need for new chemical space for the sake of high throughput screening, for the sake of for the sake of drug development. I wanna give you a brief idea about the drug discovery process as well. You see, it is established so far that we need a new chemical space, we need new molecular entities, we need new chemical entities, NCEs. But so what? I mean, once you have synthesized thousands and thousands of molecules, what is next? So once the first step in the drug discovery process is to have these compounds screened against certain target identification and validation, and then you do lots of assays, bioassays, and then you develop the hit molecules, and then you come up with the lead generation. This phase will usually take 3 to 5 years, and lots of medicinal chemists would be involved. A typical drug discovery process on a whole usually take 14 to 15 years, and hundreds of chemists, biologists are involved in that process, and it usually costs approximately $2 billion US. So after the first phase, if let's say if you start with 1,000 compounds, 10,000 compounds, in the second phase, which is preclinical trial, you will have approximately 250 compounds left. right? And then on those 250 compounds, you will perform different secondary bioassays, and then you will see their toxicity in vivo and in vitro. Then you will observe their pharmacokinetics, pharmacodynamics, and the whole process usually take 1 to 2 years or maybe more. And then only few compounds, only fraction of the compounds, like 4 to 5 compounds, will actually make it to the clinical trials. And clinical trials will be based on phase 1, phase 2, and phase 3. And the whole process usually takes 6 to 7 or maybe 10 years. And then maybe if you're lucky enough, your 4 to 5 compounds make it to the next level, you will have only one candidate to the drug approval process, regulatory approval, what we say. And this process will take lots of documentation, lots of funding is involved, and the whole process usually take 1 to 2 years. And of course, once the drug is in the market, there are post-market surveillance and etc. And sometimes the drugs can be withdrawn from the market as well if they exert certain side effects. We do have many examples in the literature available. So having said that, there are multiple sources of these lead molecules. Traditionally, plants, microbes, marine world, animal, all provide rich resources of structurally complex natural products. In the past, usually the new molecules or what we call drug have been discovered either by identifying the active ingredients from traditional remedies or by serendipitous discoveries. But now we know that disease are controlled at the molecular level, physiological level, and of course we know the shape of a molecule at atomic level is very well understood. So the things are much more sophisticated now, but still there are certain methods which is still being used, which include random screening of compounds, molecular manipulation, molecular designing, drug metabolites study, and serendipity. These are some sources from where you can find the leads. These are examples of some lead molecule available in the literature, which are obtained from the natural sources like cardiac glycosides, morphine, quinine, salicylic acid, Taxol, the famous molecule, and penicillin, cyclosporine, warfarin, artemisinin. These are giant examples which have been inspired, which have been isolated from the natural product, and lots of medicinal chemistry have been done on these molecules in order to have the maximum biological activities, safety and efficacy. For example, here you can see the morphine and codeine, which are analgesic molecules. These are some ethinyl estradiol and other derivative which are contraceptive, and some antibacterial molecule, clarithromycin, and of course the augmentin. How can we forget this molecule, the conjugated one, and of course the macrocycle like cyclosporins, etc. So I have discussed about the drug discovery process, but there are certain methods involved in drug discovery as well. These methods are, first step would be target selection, which is basically cellular and genetic targets. This step involves lots of genomics, proteomics, bioinformatics studies. And next step is the lead discovery, which usually start with synthesis or isolation, then lots of combinatorial chemistry, assay development, high throughput screening, medium throughput screening, etc. And then of course, medicinal chemistry, where our interest is. And this basically involved library development, SAR, in silico screening, chemical synthesis, and after that comes the in vitro and in vivo studies, and then clinical trials. Since our interest and today's topic is based on the MatChem, medicinal chemistry, so we'll discuss more about this aspect. You see, MatChem is basically a discipline which is at the intersection of synthetic organic chemistry and pharmacology. One should have a deep understanding of synthetic organic chemistry in order to develop methods so that the libraries of the molecules can be easily synthesized, and at the same time one should have the basic understanding of pharmacology, so that you know what kind of functional group will exert what kind of biological properties, right? So that you can have some idea about their biological activities. This would involve a focus on a small organic molecules that are usually being used in the drug discovery and what we call hits. And after that stage come hit to lead, which involve lots of optimization, and then of course process chemistry. Process chemistry is entirely different than the basic chemistry which we do in our laboratory. This involves basically scaling up the reaction. That is basically in our laboratory, basic labs, we synthesize molecules in small quantities, not in the bulk quantities, but the process chem is basically convert your reaction into large scale reactions. If anything comes at the lead molecule, you need to have that molecule in larger quantity, kilograms literally, right? For that, scaling up any reaction is not so easy. You need to carefully optimize the reaction again. So process chemistry is involved, and then of course the development. Library development is the main aspect of MatChem, which basically is the collection of a stored chemical along with associated databases. There are hundreds of databases available through which you can have the idea about the designing of the molecule. And of course, these libraries assist you in HTS and helps in screening drug target based on organic chemistry. And and then comes the SAR, that is a structure activity relationship studies. These are the extremely important studies in any medicinal chemistry process, because these are the studies which helps you to identify the pharmacophore. You know, there are certain molecules, complex molecules, which have diverse skeleton, but there is only a small entity within that molecule which is responsible for the biological activities. Everyone knows the example of artemisinin, and we know that the peroxide linkage in the artemisinin is basically responsible for the antimalarial activity. It is very well established. We know that. So these are SAR studies which make it possible to give you understanding about the active pharmacophore. And this is basically enabling you to prepare more active molecule, and this is how you basically eliminate excessive functionality from any particular molecule. This is the example I have taken from the literature, which is, you see, this is the morphine molecule, and right on the top of it is the methyl morphine, which we call codeine. Both are being used as the analgesic, but codeine is, you see, only difference of a methyl group makes it 10-fold less active. This is, 1/10 less analgesic as compared to morphine. And you see there, down there in the summary, they have given you some certain idea, like which functionality will actually increase the activity or decrease the activity or would have no effect. For example, it is mentioned here that this ring E is basically responsible for the biological activity of this morphine molecule. And it is reported that if there is no this ring E, there will be no activity. However, if we look at this phenolic hydroxy right here, if we remove this, we will have reduced activity, as we can see in the case of codeine. If we methylate this hydroxy, we'll have lesser activity. And of course, and there are some other functionality like this hydrogen. If we replace it with hydroxy, it will enhance the biological activity, and there are certain other groups as well. So this is the basically SAR study which we can take into account when we design our own molecules in our own laboratory. And then comes the in silico screening. My colleague, Dr. Riaz, will describe you today about the various tools available in the chem informatics and bioinformatics, which you can use in medicinal chemistry to design your molecule of your wish, right? But briefly, this is computer simulated screening of chemicals, and it helps in finding structures that are most likely to bind to the drug target, and certainly it filter out enormous amount of chemical space, and of course more economical than high throughput screening, but it has to be validated through the experimental means anyway. So this is the virtual screening workflow. First step is the library preparation. Second comes the filtration through structure and ligand-based filtering, which which involve similarity-based search methods, fingerprinting-based method, chem informatic filters are available, pharmacological based filtering, docking, simulation, QSAR, lots of other tools are available to filter these molecules. But eventually, once you have filtered down or narrow down your library, then it has to go through the experimental validation. And then, of course, there are so many other tools which you will be able to discuss with Dr. Riaz in second half of today. And of course, the understanding about the chemical synthesis is very, very important, because it involves production of lead compound in suitable quantity and quality. When I say quantity, it is basically related with the process chemistry. When I say quality, that means the purity of the molecule, right? Whatever you are synthesizing has to be pure, because if the compound is impure, you cannot say which part, whether the actual molecule or the impurity is responsible for the biological activity, whether they have synergistic effect, right? So you need to have good quality molecules to allow you large scale animal studies, and maybe the human subjective studies as well. And suitable drug formulation, you cannot eat a compound like as it is. You need to have certain type of formulation. It has to be bind with the excipients, so that it is if it is oral route, so it can be swallowed, or if it is injectable, then it has to be mixed with certain agents. So these are some examples of blockbuster drugs, which have actually developed through all this process which I have just mentioned. It looks very simple on a screen, but behind each drug there is a work of hundreds and hundreds of chemists and biologists, and 14 to 15 years of work, hard work, and then lots of money is involved behind. And of course, the rational drug designing, and you see, it took Pfizer about 18 years to develop this piroxicam, the anti-inflammatory drug. It was launched in 1980s in the golden age of rational drug discovery. The starting point for the development was chemistry driven, because to identify the acidic but not the carboxylic acid containing moiety. They don't want it to have carboxylic moiety, rather they wanted to have a comparable structure. So what they did, they basically installed the sulfonamide moiety right here. You can see. So this is the sulfonamide, and this is the structure of peroxicam. So it needs, several generations of leads were refined and ultimately led to the successful structure with an acceptable safety and activity profile. Beta agonist salbutamol is another example which is being used for the treatment of asthma, right? And one has to have the idea about the quantity which is required for the beneficial effect, because if the quantity exceed the required amount, then of course it can exert the side effects. or the undesirable effect. Like if we take salbutamol in larger quantities, it can lead to heart palpitation, etc. And as a medicinal chemist, we need to know certain functionality which we don't want, which we need to avoid. You see, as a general rule, we need to avoid all kind of electrophiles. Not always, but most of the time, right? For example, we don't want like acyl chlorides. These are highly reactive. We know as a chemist, it's our basic understanding. We need to avoid these type of molecule. We need to avoid these ketenes, the epoxide, etc. So these all are electrophile, and they can easily covalently bind with the nucleophile within the body. What are nucleophile within the body? These are proteins, DNAs, and once these molecules bind with the biological molecules, they can have the toxic effect. Most common of them is the hepatotoxicity. If they bind with the liver cells, right? And of course, don't forget that in the body, chemicals are metabolized, right? Because we have acidic environment in the stomach. This can lead to lots of metabolism of the compound which we are taking as the drug. So we need to consider carefully the fate of these molecules within our body, right? Will any of these metabolite will be chemically active? We need to take this thing into our account. How we can judge? We need to have basic understanding about the reaction mechanism, so that we know if there is acidic environment, what kind of functionalities will be hydrolyzed, like, or similar stuff. Okay, as a general rule, these are some unwanted groups which we want to avoid in our molecules, which we are optimizing as a hit or lead. So like electrophilic aromatics like this, chloroamines, certainly we don't want. Terminal acetylenes we need to avoid because they are very active. Epoxides, nitro compounds, and alkyl sulfonate esters, certainly we want to avoid that because they are very reactive, as almost comparable with the acid, carboxylic acid. The isocyanates, of course, the acetylating agents we need to avoid, and mask anilines. When I say mask aniline, we can see here this aniline, which is protected with this acetyl group. So this is what we call mask aniline. And hydrazines sometimes we don't want, and alkyl halides, of course, we don't want to have in the final compounds. And furenes, thiophene, these are certain molecules which we don't want. Of course, we cannot forget these Michael acceptors because they can lead to lots of other reactions. Okay, this is the example of paracetamol. When I was telling you about the drug metabolism, you see, this is the example of drug metabolism within the body. If it goes a normal phase metabolism, so paracetamol will be converted into this molecule, where you have the sulfone group right here, and it will convert into this glucuronide, and then these two molecules these are water soluble, and they can be easily excreted out with the urine, and this is the normal way. But if it goes oxidation, and this molecule, which is N-acetyl benzoquinine, if it binds with the glutathione within our body, it will lead to this molecule. This is fine as well, because this can also excrete with the urine. So it can excrete out the body. But if this thing, N-acetyl benzoquinine, react with the proteins within our body, it will lead to hepatotoxicity and will exert side effects. So that's why it is important to have the calculated amount of the drug. And this is very interesting that sometimes finding finding leads can be very interesting. Sometimes we want to enhance the side effect. We all know that side effects are bad. We don't want side effects of any drug, but sometimes it can lead to interesting discoveries. For example, this is sulfonilamide, which is very well-known antibacterial agent. And when it was established, it was exerting side effect of lowering glucose level in the blood of patient, and also it was diuretic in nature. So scientists were immediately, they were thinking about why don't develop blood lowering agent based on this molecule. So they basically developed this tolbutamide, which is a compound inspired with sulfonamide, and it is known to lower the blood glucose level. And then also they developed chlorothiazide, a diuretic agent. Again, this is based on the sulfonilamide are inspired by the side effects. So there are certain recent advantage of advances in the medicinal chemistry as well. Like there is a pool of privilege structures, right? Privilege structures are those which are, they have certain biological activities, right? And they are generally used in drug development and designing process, late stage functionalization, drug delivery, bioorthogonal chemistry, and many other stuff. But I want to discuss briefly about the repurposing and privilege structure, repositioning. So what is repurposing? As yesterday in his inaugural remarks, Professor Dr. Iqbal Chaudhary, my teacher, has mentioned that repurposing is probably the easiest way to develop certain drug, because you start with already established well-known drug and you repurpose that particular drug with another indication, with another for the treatment of another disease, right? So there are many examples in the literature available through which original drugs have been repurposed for some other disease. So this is what we call repurposing and repositioning. These two terms are basically being used in literature interchangeably. Sometimes they call it repositioning, sometimes they call it repurposing. Repurposing, as I just mentioned you, that use of same drug for another indication. Repositioning sometimes being used for the redressing of the disease, like installation of some certain functionality within the structure of the drug, and then test them for the biological activity. But you see, when we introduce certain functionality, even a hydrogen molecule or even a metal in the structure of a known drug, now the compound is entirely a different entity. It's a new molecule. It has to go through all these stages of safety and efficacy, all these stages of drug development process. So repurposing is more easy because it can pass the first step of the drug development process, right? But for the new indication, the clinical trials and the registration and etc. need to be done, right? So repositioning is based on phenotypic screening and systemic screening, robotic high throughput screening, and medium throughput screening as well. These are some of the examples available in the literature. So this is methotrexate, a drug which is basically a chemotherapeutic agent, and this was repurposed as immune system suppressant. And down there, it's a molecule, minoxidil, which is basically anti-hypertensive vasodilator, and it was repurposed as androgenic alopecia. And then there are certain other examples of anti-cancer drug which have been repurposed to treat human immunodeficiency virus. And then, this is another example, which is basically anti-influenza agent, which was repurposed to protect cardiomyocytes and myocardial injuries. And of course, this last one is basically anti-helminthic compound, which was repurposed for the treatment of adenoviruses. So diversity-oriented privilege structure repositioning is very well known, very established strategy. You start with certain privilege structure. For example, this tetrazole, it is part of that privilege structure pool, and very well known for various type of biological activities. So this triazole was introduced in a compound, and then it was a DPP4 inhibitor, and in some other case it was reported to possess CCR5 receptor antagonists, and various other examples. And based on the same strategy, an FDA approved anti-HIV drug was developed, which is raltegravir, and some other structures as well. So I still have 10 minutes, so I will quickly jump on examples of our own work. You see, a Nobel laureate says that the most fruitful basis for the discovery of a new drug is to start with an old drug. So based on this phenomena, I, under the guidance of my dear teacher, Professor Dr. Mohammad Iqbal Chaudhary, I work on various drugs, and I do lots of medicinal chemistry. These are some of the examples. I will quickly glance through these examples. So for example, this was one of our study which was reported in scientific reports, and it is basically where we have conducted medicinal chemistry on isoniazid. It's a drug, a very well-known drug, and we have actually designed dual inhibitors of urease and inflammatory markers. So why we choose this skeleton? Like we know that pyridine-based, drugs. Pyridine is basically the entity which is present in our drug of choice, isoniazid, which is anti-TB molecule, and certainly, many other drugs, anti-ulcer, anti-inflammatory, they also possess similar skeleton. So based on this rationale, we wanted to have dual inhibitors of inflammation as well as urease, because whenever you have ulcer, you have inflammation. So if there is something which can cure both the problems, will be cherry on the top. So this is isoniazid. We do very simple chemistry. We took this isoniazid and we combine it with various isothiocyanates. We have a pool in our laboratory, and we synthesize these thiosemic carbazide type molecules. So we synthesize a library of 25 compounds and screen them against urease enzyme and various inflammation marker. So you see, as far as MedChem is concerned, this part is the constant coming from the drug. You cannot change it. And this is the handle which basically give us the leverage for the diversity, right? So this arrow part is our diversity part. So we have identified lots of good inhibitors and dual inhibitors which are comparable with the standard ibuprofen in case of anti-inflammatory, and acetohydroxymic acid in case of urease inhibition. For example, this compound, you can see it is giving us better inhibition of urease enzyme as compared to standard, and this compound also. So these are some of the library. I will quickly pass because we only have 5 minutes left. So these are some other compounds which you can observe here are better or at least comparable active with the standards we selected. This is another example of my work along with very dedicated chemists. This is another drug, hydrochlorothiazide. What we did, we perform lots of MATCAM here also. We basically introduce a propargyl bromide on this hydrochlorothiazide drug, and while we do this reaction, we obtain two products. One is tripropargylated and the other is monopropargylated. We were interested more in this one. So we isolated this, and then on the other hand, we prepare a library of azides from the benzyl bromides, right, by the reaction of the sodium azide and the acetonitrile solvent, very simple reaction. And then we combine these prepared azides along with our synthesized compound, and we do click chemistry by using quark, very well known copper catalyzed cyclization, which is very well known click reaction. And then we have synthesized a library of these triazoles. As I already mentioned, that triazoles are, part of privileged structures. So after that, we screened this library for various enzymes, and we identified that these are, very potent inhibitors of alpha glucosidase enzyme. Some of them are better than the standard. Why triazoles? Of course, various biological activities are reported ranging from antimicrobial, anti-convulsant, analgesic, antimalarial, antiviral, many other biological activities are reported. And there are certain drugs, triazoles are part of their skeleton, like ribavirin, which is antiviral drug being used for HIV, and these anticonvulsant and antifungal drugs. So we identified lots of good inhibitors. Certainly, actually, all of them are better active than the standard acarbose, which was being used, and all of these compounds were new in the literature, so we are basically enriching the chemical space with new compounds. And of course, the good thing is all of these compounds were non-cytotoxic against normal cell lines. So this is another example where we were trying to couple. This is not based on drug, but we wanted to have this cell phone thing in our final compounds, in our library of the compounds. So we started with this substrate, and we couple it with lots of sulfonyl chloride. We have a library of sulfonyl chloride available within our laboratory, and we synthesize this type of skeletons. And then we tested against them, because sulfones and quinoines are reported to possess anti-cancer type activity. So we tested them against breast cancer cell lines, which we have available within our research group, MDA and MCF7, and we identified that they are very good inhibitor of these breast cancer cell lines. We perform some of the in silico studies as well, and then we identified that the compounds is basically binding through the hydrogen bonding and making some hydrophobic interaction with the proteins. And these are some of the inhibitors which we have identified, although not as good as the standard doxorubicin, but this is our ongoing work. We are trying to optimize our structures with some other functionalities which we think will be able to reduce, will be able to enhance actually the biological activities of these molecules. Although it is not as good, but it is comparable. Like this molecule, this is having IC50 of 5.3 micromolar, while doxorubicin is very high standards. It's very good drug, but still we can optimize this further. Okay, quickly in 1 minute, I want to tell you that yesterday Professor Parang has mentioned about the prodrugs. So Professor Parang is co-supervisor of one of our student as well. So this work, I have my student Aminat, she has done this work. And we are doing anti-HIV studies with Professor Parang. So at this, during this study, we wanted to have lots of derivative or prodrug of the tenofovir, which is anti-HIV molecule with the less bioavailability. So we wanted to have some compound which have better activity and more bioavailability. And it is very well established that the prodrug of TFP, tenofovir, have better bioavailability as compared to this drug as well, because this drug has this phosphate moiety, which basically create hurdles for this compound to be penetrated within the cell membrane. So we need to make a prodrug of it so that it can penetrate the cell wall, and then eventually it can exert its effect. So we do lots and lots of medicinal chemistry. We have synthesized around like 150 compounds based on this strategy. We do multi-step synthesis. We took certain amino acid, we protect it with BOC, make this skeleton. We then we treat this type of protected amino acid with various alcohols. This is the diversity handle right here. We use a variety of alcohols, and we develop a library of this amino esters, and then these amino esters were deprotected, BOC was removed, and then these esters were eventually coupled with the tenofovir, and we produce a library of compounds. So we utilize various handle. We change, sometimes we change the amino acid, sometimes we change the alcohol part, and then we introduce different diversified handles within our library. So I'm not going into a detail of each and every one, but you can see these red parts are basically our diversity handles. So based on these strategies, we have actually synthesized some 150 compounds, and they are still in the process of testing. Some of the results which we have obtained so far against the anti-HIV, we have observed that the compounds are non-cytotoxic against the normal cell lines, but they have toxicity against the HIV screening, against the HIV. So you see, our compounds are actually better active than the standard tenofovir, which has 47.4% inhibition, while our compound have 79, 72, sometimes 56. So this is again the ongoing work. Apart from this anti-HIV, these compounds have lots of other biological activities as well. For the sake of time, I didn't include all the data right here. So having said that, MATCAM still have lots of potential. One need to explore the possibilities, and we need to design. In the end, quickly, I would like to acknowledge our finances. I've been, awardee of International Foundation for Science twice, and my teacher, Sir, has a project, drug development project from Sir. I happen to be part of his team, and we also have a project from NTD, which is a global network for neglected tropical diseases. And I would like to, pay my gratitude to my dear sir, my mentor, Professor Dr. Mohammad Iqbal Chaudhary, for his guidance throughout my life. As he mentioned that I joined him when I was a child, and I still feel child in front of him. And Professor Dr. Iqbal Chaudhary, whom we are doing anti-HIV work. This is Dr. Atyatul Wahab for the biopsy. Then Professor Dr. Sabar Yusuf for anti-leishmanial activity, and Dr. Riazuddin for some of the clinical studies. This is my past and present students, a very excellent, dedicated team of students, Miss Fazila especially, Aminat, Arsalan, Rahil, they have conducted this work which I presented here. They are excellent students, and I wish all of them a very good luck in their future. With this, I would like to thank everyone. I apologize that I have taken a few extra minutes. Thank you so much for your kind attention.
Thank you very much, Dr. Hina. It was a very nice and comprehensive presentation. And you can also see some of the, comments from our participant as well, saying that it was very informative presentation and all. With this, if you allow me, can I take a few questions posted in the chat box for you?
Yes, please.
Okay. So the first question is, how can the drug discovery process period be reduced with lessons from COVID-19 vaccine discovery? Just from your experience, I think it's more of a general comment.
Yeah, I think as Sir yesterday mentioned in his inaugural speech that lots of drug repurposing was taking place throughout the world. Scientists were, they were taking the advantage of available pool of drugs, and they were actually repurposing it for COVID-19. So this is probably the best strategy which one can use to reduce the time required for the whole drug development process, because you start with already established drug. You don't need to go for all the safety and safety studies. Of course, you need to conduct efficacy trials, but this is how you can reduce the time spent. Chat box.
Right. So there is also another question. What will be the effect on new therapeutic targets from the natural resources other than the medicinal plants, such as venom proteins?
Effect against what?
I think what I'm not sure. Like what I think he's asking is that can venom proteins be used as new therapeutic targets? Like you have medicinal plants and their products are used as, as therapeutic agents.
What will be the effect on therapeutic targets from natural sources? I'm not really sure about the question, but I think one need to validate the target, like if they are using venom proteins. So there is, I think, lots of research going on the venom proteins, and then eventually they need to optimize various structures to see if it is binding. But I'm not really sure about the answer of this question. I'm sorry. Hello? Hello?
हाँ जी पूछना please. Hello, Assalamu alaikum, Dr. Hina. I was just asking about the availability of Professor Iqbal so that we can introduce and welcome our guest speaker. Thank you very much for taking question and answers. And it was, again, a very nice presentation, educative and, wonderful.
Thank you so much, Karima. Thank you so much.
So for our participants, we will have our next speaker from China, Professor Dr. Shang Wang. The title of his talk today is Examples of Medicinal Chemistry Research, Small Molecule from Chinese Herbal Medicine. And with this, I think, let me just wait for Professor Iqbal, if he would like to introduce our guest speaker. Okay. Dr. Hina, Dr. Iqbal would want us to continue with it. He has a meeting. So should I introduce the speaker or would you like to do that?
Yeah, this is fine. I can introduce, no problem.
Okay, please. Please welcome and introduce our guest speaker, Professor Dr. Shang Wang.
Okay. So again, it's my honor and privilege to introduce our next speaker, who is Professor Dr. Shang Wang, who is professor at State Key Laboratory for Chemistry and Molecular Engineering of Medicinal Resources, School of Chemistry and Pharmacy, Guangxi Normal University, Guilin, China. Dr. Wang is a professor of bioorganic chemistry and deputy director of State Key Laboratory for Chemistry at Guangxi Normal University, China. He is also the chairman of a special primary foundation for the National Key Basic Research Program of China and National Natural Science Foundation of China, and a member of Chinese Chemical Society and distinguished expert of Guangxi, China. He got his BS in plant physiology, MSc in phytochemistry, doctorate degree in biochemistry, specialist in polypeptide biochemistry at the State Key Laboratory of Applied Organic Chemistry, Kanzu University of China. Dr. Wang got the Science and Technology Award of Guangxi 7th during 2012 and 2016, respectively. Currently, Dr. Wang's research focuses on therapeutic basis of important medicinal resources of Guangxi, natural compounds derived from traditional Chinese medicine, studies on anti-cancer mechanism of apoptotic pathway targeted natural product like compounds and their derivative. He published more than 180 papers in organic matter, green chemistry, ChemCom, bioconjugates, chemistry, Journal of Medicinal Chemistry. These are the prestigious journals in the scientific world. So he has also obtained Chinese patent authorization. With this introduction, I request Professor Wang to start his presentation. Over to you, Professor Wang. Professor Wang, can you hear us? You can start.
Camera be off.
Professor Wang, can you hear me?
Yeah, can you hear me?
Yeah. Now we can hear you, but we cannot see you. I request you to kindly turn on your camera. Okay, excellent. So with this, sir, you can start your talk.
Yeah. Good morning, everybody. Great thanks to Dr. Hina for give kind introduction of me in detail, and great thanks to Professor Iqbal and organic community from comstech and ICCBS for the invitation. Today, I want to introduce some progress in my group about pharmacochemical study on your ethnic medicines. My presentation include 4 parts. My 4 parts and 13 examples in relation to natural medicine studies, structure modification, multi-drug resistance, and immunological effect of small molecules, respectively. It's clear that nature will provide the continuous source of new drug leads. Efficient drug development depends on chemically organic synthesis from core study, including new natural product discovery and diversity-oriented synthesis to establish the natural product like the library, share the benefit of innovation from biodiversity to structural diversity to functional diversity to achieve the efficient transfer from a resource to leading compounds. Natural products. Guangxi is a subtropic mountainous area located in the southwest of China, has plenty of natural resources where live the Zhuang, Yao, Dong minority peoples. We focus on our attention on the herbal medicine used by Yao ethnic group typically the 104 species. Part 1, Nature Medicine Studies. Let's take a overview of natural product types that include the organic acid, alkaline, neutral, and aliphatic compound. For example, phytic compound, alkaline, terpenoids, sterols, and acid. The structure and the group affect the polarity and the chromatographic behavior of a compound, so different separation method are needed. This slide shows the common methodology to initiate the column chromatograph separation and purification. Pre-separation treatments are needed by using microporousing, followed by repeat refined separation. Use silica gel, reinforced silica gel, linseed gels, sulfadex gels, monitored by PLC, followed by repeat analytical and prospective HPLC installed ODS column and the UV detector. The CD spectral with the CD detector will be used for chiral compound, and the optical rotation are needed to be determined and followed by a comparison of the experimental and calculated ECD spectral to determine its absolute configuration. What we needed to emphasize here is that for each separation, the peak of UV time must be detected by HPLC instrument to check the purity and the content of the separation fractions, or check by TLC red color region to show the different color spots represent different type of compound. Or if aimed for the active compound, active guard separation are recommended. As for structure determination, UV spectral, IR spectral, high mass spectral, NMR spectral include 1D or 2D spectrals, and single crystal diffraction might be used for identification of a new compound. For optical rotation detection and the CD spectrum, this show the comparison of the experimental and collected CD spectral and the determination of observed structures. For active screening and the mechanism investigation, the methodology are listed as follows: enzyme assay, cell availability assay, flow cytometer, molecular docking, SPR experiment, DNA binding study, real-time PCR assay, Western blot assay, immunofluorescence staining. And considering the active screening for anti-cancer constituents in herbal medicine, the active target was chosen by the latest development in the pharmacological mechanism and the new target discovery worldwide. They are listed as follow: for the apoptotic pathways, anti-angiogenesis, cell cycle arrested, anti-drug resistant, anti-migration, and metastasis autophagy immunology method. And the typical apoptotic mechanism include mitochondrial pathways, endoplasmic pathways, and cell receptor pathways. First example, the Antibacterial medicine, Yan clinically used the anti-HBV and liver cancer. The prescriptions as total alkaloid injection used by clinical for anti-HBV. And activity guide separation of the extraction of this aeropath. Fraction in isoacetate part was most active against the cancer cell lines, and theory of lignin amide compound, including 3 ones, was isolated in this part, together with a theory of isoquinolines. This compound number 1, 2, 4 share the same skeleton. However, in activity screening, we can see that compound 1, 2, 4 are different activity. Compound 4 have the most activity among them. So compare the structure. One substitute at group R1 and group 4 with methoxyl group will reduce the activity. However, in compound 3, 6, 7, they have a same skeletons, different in the steel structure at carbon 1 and carbon 2. This absolute configuration is the size. It has the most activity. The compound 3 named choline C. To summary, the choline C possesses the most potential inhibitor effect against the MGC cells. SAR analysis show that steric and chirality of the lignin plays a crucial role in the pharmacology-relevant events. Choline C may induce the apoptosis through both intrinsic and extrinsic apoptosis pathways, accompanied by downregulating the expression of BCL2 and fast ligand in a time-dependent manner. In addition to the separation, we found that 5 new monotyped endonucleotides in another Yao medicine, Mapianthus idos. The alkaloids show the mechanism of inducing tumor apoptosis and arresting the cell cycle. Part 3, structure modification. Actually, in most cases, we found the natural products with normal structure usually have a low yield and a high content of compound. Normally, they have a poor activity, which restricts their application. Structure modification will be needed, and the methodology as follows. That was mentioned just by Dr. Hina. So let me briefly introduce the principle. The first, for the structure design is according to the Lipinski rule of 5 for the judgment of drug-like property. Suggests the desirable structural features. that is the molecule weight less than 500 Dalton. Hydrogen bond donors no more than 5, hydrogen acceptors no more than 5. Log P value less than 5. For rational drug design, more called redesign based on the pharmacophore, and we also wanted to design the dual targeting or multi-targeting drug by combination the targeting structure fragment and do some organic synthesis for conjugations. For structural optimization, aim for refined ADMT properties. That's what the good absorption, the better distribution, and metabolism, and execution, and low toxicity can be predicted by some software to optimize the solubility, the targeting properties, and the selectivity to reduce the toxicity. And the structure activity relationship will be very important to do the QSAR. For example, when we do the modification of pentalic cyclic triterpenoids, we found in reference that the activity may be affected by the modification at ring A and carbon number 11 and carbon number 28. So most modification were taken at this stage, and pharmacinic and pharmacodynamic evaluation and the toxicity study have followed. After the in vitro study, we also need to carry on the in vivo study. That means to establish the animal experiment by using mice or rats. For side chain group introduction, we often chose aminophosphonate and amino acid. For aminophosphonate APA, bearing the NCP fragment, nitrocarbon phosphonate fragment. That is the biosensitivities of amino acid function include maintain the capacity of forming network of hydrogen bonds, negatively charged organ atom around the phosphonate atom, good mental contain complication property, possible creating high activity, selectivity, inhibit targeting catalytic protein classes, for example, for targeting different peptides, FFP synthesis. Different type of NCP fragment targeted key enzyme in the amplification and metastasis process of cancer cell. Meanwhile, the ethnic group target the phosphodiesterase, which was found overexpression in cancer cell. Amino acids such group have amphoteric properties. Functions include maintain the physiological pH and hydrogen bonds, change the drug solubility and the metabolism pathway, enhancing drug activity by targeting the key enzyme, reducing drug side effect or vice reactions. More important, when a clinical drug, Pentosacin, was linked with the serial acid side chain group, the cytotoxic increased near 40 times at the meantime. Phenylserial moieties plays a key role in active the fast pathway. It throws a light on the choice of a natural molecule for functional pharmacophore in new multi-targeting drug development. As an example, many anti-cancer clinical drugs contain acetylquinoline scaffold with the chiral side chain group. Introducing a polar side group was also a common approach in quinoline structure modification. Rin is the main component from polar group caspitotum. As a clinical drug, it was used for osteoarthritis therapy targeting RXR. The results show that Rin APA conjugate have obviously enhanced inhibitory activity of cell amplification in compared with the Rin and refined DNA bonding property and induced HTC clone cell apoptosis, arrest cell circle in S and G1 stage with large proportion of scales. For another example, alizarin is a main component from Robia cordifolia. Alizarin APA conjugate was designed using carbonic linkage and diorthophosphonate prodrug group, inspired by the structure of HTAC clinical drug. The structure, its HTAC inhibitor, SAHA, was selected sim long linkage and sim property terminal group, and in the conjugation of original molecular design. From the illustration, a bonding model of a compound with the HTAC shows the phosphonate atom obviously close the mantle zinc cation. When chiral carbonic acid linkage was used in conjugation, it was found that to have 2 pairs of diastereoisomers isomers and 4 enantiomers, the separation crystal was detected 2 and 4 times. Activity increased. Different enantiomers act a different manner. Separation, the pure enantiomers act in different manner mechanism, and ADMET prediction listed here predict this conjugate have a drug-like property and with a low risk of efficient. Constant study, including ACUSA study, including COFA and CONSA analysis, predict show that some group need to be increased. However, further modification show that only limited improvement can be obtained. But the effect of DNA bonding and apoptosis-induced induction was preserved. Pine tree is TCM rich in pansy resin is diterpenoid as the typical main component. Dehydroabiatic acid, DHA, enhanced the inhibitor effect of anti-cancer drugs to the cervical liver breast cancer cell through an ion channel mechanism. However, it has a poor solubility and activity, and its active mechanism is unclear. So we design and synthesis 3 series of DHA also Euro API conjugates and 3 DHA Euro amino acid alien derivatives with oxo or oxyamine substitution. It is found that the activity improved significantly. The mechanism of study showed that this compound could induce the cell apoptosis through the loss of mitochondrial membrane potential, enhanced the produce of RS and cytochrome C, elevation the BCL expression and down-regulated BCL2, and active the caspase 9 and the caspase 3, and the drug-like property was also improved. Moreover, there is a low risk in druggability by source-wise prediction evaluation. A step forward, we think about another NCP fragment and synthesis DHA biphosphonate derivatives. The cytotoxicity was found to be excellent, with IC50 is near 1 micromolar, induced apoptosis through mitochondrial pathway. Excitingly, it was found that the FAST was active. The cell circle was arrested at the G1 stage in large proportion. So we expect that the CDK may be involved in this process. The compound was detected to have CDK2 inhibition with IC50 value 130 nanomolar. Binding model show that this compound could bind to the ATP pocket sites in the same way as the natural ligand binding, except as an expected DHA scaffold have certain binding effect to CDK2. Part 3, anti-tumor multi-drug resistance. This process involves much complicated cell signal pathways from EGFR to EGF to MMP to UPA to PGB, and affect to produce the MDR effect. Lysematia contain terpenoids, saponins, and flavonoids have anti-cancer activities being used for treatment cold and headache, for treatment blood circle and skin diseases, lungs and brain cancers. 14 Olinen type new terpenoid saponin was isolated from air part of this herb, and the 7 saponins contain the glycol cyclamine rating A, except the moderate cytotoxicity against the human cancer cell lines. Simultaneously, 5 of them have an inhibitory effect on drug-resistant lung cancer cell lines with resistant factor of 0.9. Solubility Melidolphy is a famous TCM used for hepatitis and skin diseases. Isolation, a lot of compounds, for example, flavonoids, biflavonoids, lignans, and pyrrolocaloids. References show that phenolic compounds are potential candidates for MMP inhibitor. For example, the lignan and biflavonoids. We obtained a theory of a new lignan and biflavonoid from this herb medicine. Activity screening or mechanism study with research show that some of them have inhibitor effect of test cancer cells and have the effect of inhibiting drug resistance and tumor cell migration by targeting MMP. This is the targeting model. A step forward, another species of Salagilla genus, Salagilla tamarissima, from famous TCM, has been traditionally used in Asia for treatment of hematuria and annuals. It extracts inhibited receptor activator for NF-kappa B ligand induced osteoclastic diffractions, inhibit breast and cervical cancer. Biflavonoids regulate the expression of MMP and have a SEH inhibitor effect. Salagillin have an effect as insulin mimic PTP inhibitor. Salagillin represents the structure intriguing class of phenolic pigment featured unique organo and crino method functionality, which have been found only in the genus Salagilla. In our recent study, a Salagillin new compound named Salagitarimolin A normal Salagillin analogous featured the unique motif of in the center of benzopyran, and another new Salagillin analogous together with 8 known ones were isolated from this herb. Compound 1 showed remarkable inhibit against the phosphodiesterase PDE4 P2 with IC50 value of 40 nanometer, being 20-fold stronger than the positive control Rolipram. Furthermore, compound 1 significantly inhibits the tumor polymerization. We performed the doping study for this compound to elucidate the structure basis. Surprisingly, the common compound of binding site is similar to the colchicine, a well-known tuber polymerization inhibitor. Compound 1 obviously inhibited polymerization and was observed at 2 concentrations, and the rate of assembling as well as the final amount of microtube was lower than the control. Moreover, we have also obtained a series of new structural phenolic compounds which have an effect of inducing drug resistance and drug tumor cell migration by targeting MLP. Another story, we are concerned with the process targeting NF-kappa B. NF-kappa B controls the transcription of DNA and cytokine production and cell survival. Incorrect regulation of NF-kappa B has been linked to cancer. In this study, we isolated a series polyhydroxyl substitute pentalic triterpene from Euphorbia cyberdenia. Polysubstituted hydroxyl group at ring A have the most activation, and it acts as the blocking ROS-NF-kappa B pathway, and the effect shows the effect in the following pharmacological relevant events. Further step and another choice of the targeting enzyme is that HPS 90. When DNA damage in a cell, it will go through PAP to P53 to P24, got to cell circle arrested and apoptosis to go to death. However, has the 19 in cancer cell can stop this this process. Hyper-19 in cancer is stabilization of mutant protein, and it can act as a protector of less stable protein produced by DNA mutation. So Hyper-19 inhibitor will help to identify and eliminate mutation in cells, and terpenoid can be a candidate. We started from glyceretinic acid. Glyceretinic acid is the main component in the TCM. Started from this as a material, we do some by ideal side group linkage. We synthesis a series of conjugate derivatives by introduction of heterocycle side chain group. Derivatives show that a strong Hyper-19 inhibitor affect the apoptosis side circle and inhibit the MDR and cell migration. Further, we also do some conjugate by using acetic acid conjugate triazole, and the derivatives blocking NF-kappa B activation and inhibit cell migration and have the effect in the pharmacological relevant events. Part 4, immunology effect of small molecule. Immunotherapy have been a hot issue of the last 2 decades. Immune checkpoint inhibitor have been documented as the breakthrough in cancer immunotherapy. FDA approved series of monoclonal antibody as anti-PD-1, anti-PD-L1 agents have shown clinical success in cancer immunotherapy. However, only a fraction of the patients with advanced cancer respond to them. There is a strong demand for new immune anti-tumor agents. There is still no approved clinical small molecules in this aspect. New strategy combined therapy and immunotherapy would enhance the clinical benefit due to the simultaneous targeting the different tumor cell survival pathways and overcoming drug resistance. For example, platinum drugs incorporate ideal or immune checkpoint inhibitor to have achieved clinical therapy signaling. We want to settle our strategy by combining the chemotherapy and immunotherapy effect in small molecule. Either one is oxidoreductase catalyzed tryptophan catabolism. Its overexpression results in the overconsumption of tryptophan and the suppression of T cell response and immune tolerance involved in tumor escape immune surveillance. STAT3, a transcription regulator of diverse tumor promoting effect, is constitutively active in both tumor and immune cell with the tumor environment. STAT3 regulates the multi-gene crucial for tumor development. Active STAT3 inhibits the expression of immune activation modulator and the promoter of immune suppressive factor. We re-examined the structure of cytotoxic drug contain anthraquinone scaffold. Literature showed that quinolines are cytotoxic and immune-related targets, which are conducive to the simple method realizing the biofunctional anti-tumor agents that integrate cytotoxic activity to facilitate the tumor shrinking and the immune mechanism to eliminate the residual cancer cells. We designed 3 series of naphthoquinone oxime structure activity relation show that the key feature of the structure, hydrogen bond acceptor is necessary, oxime is necessary, and the additional also amide to oxime will reduce the activity. So the second section show the best activity. PD-1 inhibitory and cytotoxic activity of this compound show the similar trends in corresponding to the in vivo effect. Docking study and the SPR assay show that this compound binding IDO1 and STAT3 with the KD value of at 0.5 micromolar, and for in vivo animal models establishing we chose liver cancer and melanoma for the cost theory necessity. The compound show that good in vivo therapy effect on liver cancer and melanoma in mice. As a conclusion, expanding the use of natural products from traditional Yao medicine and the modern pharmaceutical research have active value. More application examples are expected in future. Thanks for the initial support and my group. Thank you very much for your attention. Wishing you health and life and successful working. Looking forward to meeting in Guilin in future. Thank you very much.
Thank you very much, Professor Wang. It was a very informative and interesting presentation indeed. With this, I would request our participant to please post questions in the chat box. Meanwhile, I would request His Excellency Professor Dr. Iqbal to say a few words on Professor Wang's lecture.
Dear,
hi, Professor. Hi, Professor.
Thank you so much, Professor Wang, a very dear friend of ours. It is a great pleasure of listening to Professor Wang. He is known to us since last several years, and he is a very active scientist and an excellent human being. I always enjoyed meeting him It was always a pleasure of meeting him in Guilin and in Karachi. And we were always very, very happy because he is very innovative and he's expanding his research in various areas. So he was able to link natural product chemistry, traditional Chinese medicine with synthetic chemistry and organometallic chemistry. So I was very pleased to listen to his lecture. It was a very good quality science, and he presented them in a very nice manner. So I'm sure that all of you have benefited from rich experience of chemistry and linkages of chemistry with the discipline of pharmacology in the presentation of Professor Wang. So thank you very much, dear friend, Professor Wang. It is
Thank you.
Thank you.
Very nice. Nice to meet you.
Thank you very much, sir. And thank you to Professor Wang as well. With this, we will move on to our next speaker. Our next speaker is Professor Dr. Cheng Lu. The title of his talk is Development on Potential Metal-Based Veterinary Drugs with Improved Efficacies than Fluoroquinolones. And with this, I would request His Excellency, Professor Dr. Iqbal, to kindly introduce and welcome our guest speaker.
Thank you, Hazima.
Thank you.
Thank you very much for providing me opportunity of introducing yet another very good scientist from our friendly country, Professor Cheng Liu. Professor Liu is also known to us.
Liu, thank you. Thank you.
Twice in Guilin, and I was also very pleased to see his research group moving forward with lots of great ideas. He's a young faculty member, a very good addition in the center in Guangxi Normal University, and certainly a very good finding for seniors to have a young man of his caliber. Professor Liu was born in 1977, so he's a very young man in Hebei province of China. He received his bachelor degree from in engineering from Tianjin University. Tianjin University is a very prestigious institution. And master degree of engineering from Beijing University of Chemical Technology in 2003. He received his PhD from Nankai University, again a very prestigious university. And in 2007, he was then employed by the Guangxi Normal University, and since then he's working with a group of very senior scientists. In 2008, he was moved up to an associate professor rank and chosen as tutor for graduates. Then he finished a postdoctoral program following 2 years. In 2014, he was then moved up to professorship. So he became a professor in a very young age. His research interests mainly include inorganic medicinal chemistry and bioorganic chemistry. And he has developed several new metal-based anti-cancer agents and veterinary drugs. To his credit, he has several publications and many international patents. Along with this, we know that his research involves lots of modern aspects of medicinal chemistry, and we are eagerly looking forward to your presentation, Professor Liu.
Thank you, Professor Chaudhary. Thank you.
Thank you very much.
So I would like firstly to make sure that can you hear me clearly? Because I need to confirm if the Wi-Fi is stable enough.
Yes, Professor, we can hear you very well.
Okay, thank you. Then I would like to share my screen now. Okay, thank you for the introduction from Professor Chaudhary, and also thanks the workshop to give me this opportunity to partly introduce my research work in the past several years. And now, okay. So now my topic of the speech is mainly about the development on potential metal-based veterinary drugs with improved effectiveness. So this is some special topic from the other researchers because most of our research mainly to get some medicines or potential drugs for the human beings, but we cannot ignore the other big family of animals. So since about 10 years ago, I tried to study and make exploration on the veterinary drugs. And so here I firstly introduced two kinds of fluoroquinolones named marbofloxacin and enrofloxacin, and to introduce what I have made on the metal complexes of these two drugs as medicinal ligands. Okay, so my speech mainly could be divided into 4 parts, and the mainly is section 2 and 3. And I want to make the listeners to understand that there's some difference between the metal complexes of marbofloxacin and enrofloxacin. Although their coordination structures seems similar, but their aim and targets and the efficacies are some different. Okay, so first, the section 1 will be a brief introduction for the veterinary drugs. And we all know veterinary drugs is also we called animal drugs. They were broadly used in many aspects. The mainly purpose is to prevent, treat, diagnose the animal diseases. And sometimes we also need some called veterinary drugs to purposively regulate their physiological functions. So sometimes we also call them the medicated feed additives. So it's some lack of nutrition for the animals. And on the other hand, in China, I'm not sure if it is also popular in the world, that in China, some small families of medicine also were included in the veterinary drugs, like for fish, for bees, and for silkworms. So it's a large family. And so the animal drugs or veterinary drugs, I think they should be mainly served for the livestock, like the cows and the chickens, and like we cannot forget another family of pets, like cats and dogs. And I think the pet medicine should be a sunrise in the industry, because I think with the living improvement of the lives in many countries, more and more people, not only the elder people, but also the young girls and like the children, like to herd the pet in families. And so the pet medicine should be focused on in the future, is my suggestion. So the veterinary drugs can be divided into many varieties, and generally were 4 classes. The first may be more familiar for the biological researchers, like vaccines, like the probiotics. So this is not our main topic here. And the second is chemical drugs. This is maybe the most predominant families for veterinary drugs. So it's not only including the antibiotics, although antibiotics should be the main branch, but also other like intermediates or some semi-synthetic drugs, or like some, you can recall that like the sulfur amides, like this. And the third branch should be the Chinese medicinal materials, and some of them are Chinese patent medicines. That means a combination of several Chinese herbs together, and then to make some multiple functions. This and mainly used in China. And so how to introduce it scientifically to the worldwide is still a problem, because it's not easy to understand the multiple function and the complex. And generally, the fourth branch include the other biochemical drugs, and we also have to skip it, and due to the time limited. Okay, we back to the background of what I want to make the study. Since 2010, about 10 years ago, China has started the agriculture modernization strategy. So there will be more and more modern farming and animal husbandry to be produced or grown in China. And so there will be accordingly a rigid requirement for different veterinary drugs. So after reading that policy, I make my decision to have some explorations on this field, in this field. And after some investigations, I noticed that most of the newly approved veterinary drugs, not only in China, I mean that worldwide, should be chemical drugs. And many of them actually were generic drugs from some very well-known parent structures, parent skeletons. So they mean most of the new drugs were derivatives from some known drugs. So as an inorganic chemist, not only to myself, but to us all, I think so. The metal-based veterinary drugs, actually, they are metal complexes of some medicinal ligands, should also serve as a kind or a class of veterinary drugs derivatives, because not only the organic derivatives, but also our metal complexes is a kind of modification or some decoration on the parent skeleton. So why not? And then we focus on the second branch of the veterinary drugs, that is the chemical drugs. And in the chemical drugs, I think the most famous should be the antibiotics, that they were originally produced fully or partly by some microorganisms. So they were divided into a series of classes. And the predominant ones should be macrolide, like this, and the beta-lactam penicillin is much more famous to us, and the aminoglycoside, like the streptomycin, and like this. So antibiotics is a predominant branch, but on the other hand, this branch has been heavily or severely limited in recent years, especially in Europe, in America, and now in China. So the use of antibiotics in animal as veterinary drugs will be more and more hard. So we have to switch it to other branches. Now I would like to introduce another big class named quinolones. Quinolones drugs are also well known to the chemists, and it can be classified into 4 generations. And let's skip the first and the second and jump to the 3rd generation of quinolones. And most of the names for the 3rd ones are famous enough, like the norfloxacin, ciprofloxacin, and enrofloxacin. I need to notice that enrofloxacin was specifically used for animals. That means it is a pure veterinary drug. And then in the past decades, the 4th generation quinolones, also floral quinolones, were developed, like the moxi, spa, or levofloxacin, and they spread their antibacterial spectrum to both the gram-negative and the gram-positive ones. So this is about quinolones. And here I would like to introduce a new 3rd generation one named marbofloxacin. And it's some like enrofloxacin. It is some new veterinary drug and to specifically used for animals. Okay, so the second section, I would like to introduce why I started my research on the metal complexes of marbofloxacin. Firstly, let's get to know it. Marbofloxacin was first produced by Roche in Switzerland, and in 1995, it was primarily launched in market in Britain. And it showed a broad spectrum antibacterial activity. You can see from the MIC values below towards most of the typical bacterial strains, and we can find most of them were lower than 1 microgram per milliliter. So it is higher antibacterial enough. And it also showed fine pharmacokinetic properties. So the enough reports have suggested that the marbofloxacin can effectively penetrate into the tested animals on their main organs. And so it also showed high bioavailability and enough long half-life of elimination. And we also noticed that from this report, and what was tested was mainly about animals of the pets, like the test on the dogs and the cats. And otherwise, there will be some livestock with high value, just like cattle. So that means there's a small problem we have to face to marbofloxacin. That is price. Marbofloxacin is a new veterinary drug, quinolones, with a high price. In US dollars, I think should be about 600 US dollars per kilogram. So it about 20 or 30 times than the normal 3rd generation quinolones. So this is the main problem. So this is why it was focused on the pets and the high-value livestock, because to cure the disease on them, the cost could be partly ignored. And this is the crystal structure we got. And on the other hand, the marbofloxacin also showed satisfying toxicity. Its toxicity is fairly low, and no mutagenicity and no dominant lethal were found, and also no obvious reproductive toxicity, only with some slight tetrodogenicity on the fetal mass. But it also showed some deficiency. The first is the poor water solubility, especially when it was made in solution for intravenous injection. So even in warm water, the marbofloxacin is also only slightly soluble. And on the other hand, we also noticed that the LD50 value for it is 1.1 gram per kilogram. This value may be some low toxic when comparing with some cytotoxic toxins. But for a veterinary drug, it also means the toxicity is obvious. So how to decrease it is also another problem. And as a recent development in China that we found about 5 years ago, for the new veterinary drugs of marbofloxacin were both approved by China. And actually, they are not showed any, sorry, let me see, is the Wi-Fi problem? Is okay? Okay, sorry. And improvements, they are just served as marbofloxacin API. That means bulk.
Professor Hang, we lost your screen. Professor Hang and Cheng, we lost your screen. Are you?
Dr. Henna, I think we have lost him. He's not in the list of the participants. I cannot see him.
Yeah, I think so. What shall we do?
Can you ask Jan if she has his number?
Okay,
WhatsApp?
yeah. Let me do that. Okay, he's back. Dr. Liu, can you hear me?
Yeah.
Yeah, we lost you.
Sorry, can you hear me now?
Yeah, yeah.
It's okay?
Yes, it's okay. You need to share your slides again.
Okay, no problem. I think he's here.
Yeah.
Sorry for that.
No
Okay,
problem.
okay. I will continue. And so that means the new approved veterinary drugs on marbofloxacin is mainly about its bulk drug or tablets. So no chemical modifications on it. So we have the confidence as a chemist to change something. So the primary research is actually very straight and clear. And I think it is simple enough for most of the inorganic chemists. You just choose the marbofloxacin as a medicinal ligands to coordinate with different kinds of metal ions, and then to screen their structure, their antibacterial activity, and acute toxicity. So our primary target is just to develop a new kind of potential veterinary drug with independent intellectual property right. And then when we choose the metal ions, firstly, we think about that the limitation of the heavy metal ions for the drugs. So the general transition metal ions was not primarily chosen and considered. So we choose the series of lanthanide, and another is a calcium. We also know that to get the metal complexes of this series. And then we firstly to get the lanthanide complexes, and almost most of them have been achieved. So this is their structures. And from these structures, we found that they almost share the same coordination mode. All of them are 8 coordinated and with the tetragonal antiprism coordination geometry. So they're almost the same. And this is why we can start one and to achieve a series. And then we also get the calcium complex of marbofloxacin. To be honestly, it's not so easy to get the calcium one because some like the sodium, so it only to form the salt, but not complex. So we need to confirm it. So when we achieve this crystal structure, we also examine their mass spectrum, and they both to prove that the coordination is rigid and is stable, not only in solid state, but also in solution. And then we think, why not try the magnesium one? And so we got the magnesium complex for marbofloxacin, and it gets a more regular 6 coordinated structure. And then another student of mine also got the other series of the transition metal complexes. And so we almost get most of the metal complexes what we want, especially when considering the metal ions. So we move to the antibacterial activity screening. This is the first, the MIC, the metal complex. The results indicate that after coordination to different metal ions, the MIC values or the antibacterial activity was retained. There are no significant improvement, but no decrease. They almost the same. And then we move to MBC, almost the same. Generally, the MBC values should be some higher or larger than the MIC. So they share the very similar results and indicating that towards most of the typical bacterial strains, the coordination of the marbofloxacin to the desired metal ions will not change the antibacterial activity. If we notice some specifications for some drugs like the quinolones, it always told us that when you use the quinolones, try to prevent to take it simultaneously with some metal ions like aluminum, like the calcium. Otherwise, they will precipitate the quinolones and to decrease the efficacy of them. And now we try to prove that stable coordination won't change it. And then we do the acute toxicity evaluation on mice. And this is the experimental conditions. Let's skip it. Okay. And then most of our compounds were tested and the acute toxicity was achieved. And firstly, let's see the marbofloxacin. And its LD50 value is about 1.29 gram per kilogram. So from this value, we can say the test conditions is reliable and scientific enough. Because you may notice that in the introduction, the official value for the LD50 value of the marbofloxacin is 1.1. And what we have got is very similar to that. And then we tested the calcium complex of marbofloxacin, and the value is 3.19. So it's about 2.5 times higher, than the marbofloxacin. That means the acute toxicity was effectively reduced after it coordinated to calcium. And we should notice that all the test was based on a one-time oral administration. So all the complexes will make it in solution and orally administered to the mice. And the maximum volume should be only 2 microliters should be that. And then you can see the lanthanum of the marbofloxacin, and its LD value is about 11. And then all the other lanthanide complexes, I did not show it here in detail, just showed fall in the range of 5 to 15. So that means all of the lanthionide complexes, their acute toxicity should be much more lower than marbofloxacin. And especially for the lutetium complexes of marbofloxacin. And its LD50 value is higher than 15. That means this is the maximum oral administration volume. We cannot get higher. So in detail, my students tell me if we give more than this, and maybe the mice will be fed to death by water. So that means it is really non-toxic. And then the other panel of metal complexes was about the magnesium and the 5 transition metal complexes. And all of them showed a much lower toxicity than the ligand. We can see that. And so like the magnesium complex, it was firstly reported. And then after that, we also do the histopathological examination on the main organs of the dead mice. And we want to see the mainly problems on them. And this is under the treatment of the marbofloxacin and many images. And so I just select some of them to present here. And for the ligand, you can see it is about the heart of the mice. And we can see that the myocardial tissue kept well, no necrosis and no hyperemia. So only slightly irregular arrangement was found in the fibers and near the cells. So it is actually low toxicity. And this was selected for the liver tissue towards the calcium complex of the marble. And we can see also the liver's tissue structure kept well, no necrosis, and only slightly hyperemia was found in the central veins. Yes, you can see that. And then is a lanthionide complex, diprosium complex of the marbofloxacin, and on the kidney, so almost the same. That means most of our metal complexes did not show further injuries on the main organs of the mice than the marbofloxacin. And this is towards the spleen. And we can also notice the tissue is normal. Okay, and then according to the standard classification by the SFDA, all the chemical compounds could be divided into 6 classes of toxicity. And the number is larger. That means it's much more toxic. So most of our complexes fall in the level 1 to level 3. And from this classification, we can conclude that the marbofloxacin should be a low toxic drug with level 3 toxicity. And the calcium complex of marbofloxacin also classified into level 3, although it is actually some lower toxicity than the marble. And the other lanthionide complexes and also other like the transition metal complexes all classified to level 2. They were also called practically non-toxic that generally represent to the very low toxicity compound. And for the special lutetium complex of marbofloxacin and its LD50 value won't be lower than 15 gram per kilogram. So it is belong to level 1 compounds. That means it is non-toxic. Okay, and then we move to the second procedure. So it's like a further study on enrofloxacin. So why we choose enrofloxacin as the second procedure is actually about 3 years after the study of the marbofloxacin. So this is also the life from the life or from the industry. And when I got the Chinese patent of the metal complexes of marbofloxacin, I was excited and to be confident that it will be a good potential veterinary drug to replace the marbofloxacin. But when I contact with the enterprises, and they told me it is too expensive. So when we do some research in the field of veterinary drugs, maybe the primary thing we need to notice is about the price. So marbofloxacin is really seldom. Very few metal complexes were researched on it, but it is too new and too pricey. So what the metal complexes, although they showed good perspective, but who can use it? So now I did not give up. In the past years, I still struggling and to make it as a pet medicine. That means to ignore the price. So it's also under progress. But for a more predominant veterinary drug, what we need to do is to change to a much cheaper ligand. So we focus on the enrofloxacin. And the enrofloxacin is much more famous than the marble. And it's also as the brother of the ciprofloxacin, because ciprofloxacin is just the metabolite of enrofloxacin. That also gives the enrofloxacin a good aspect that it can serve a longer term. Yeah, that means as an original drug, it works. And as a metabolite, it also works. So this is why enrofloxacin is so popular in the veterinary drug field. So in the other can change this is still a problem in the next step. Yes, so this is the problem. And the other aspects, we can see that the enrofloxacin is good enough, just like the marbofloxacin. And it was toxicity. And what we have examined is some different to that, but very clear, very close. Okay, so as like the synthetic procedure, we also get a series metal complexes of enrofloxacin. And this time we just ignore the lanthanide complexes because some of them have been reported. And now we more and more think that the calcium complex and the magnesium metal complexes should be more promising. And the transition metal complexes will not, as we thought, will be more toxic. So, and from this structure, you can see that the calcium complex of enrofloxacin was some different to that of the marble because it is 8 coordinating and only with 2 enrofloxacin on it, and the other 4 positions were occupied by the water, and so it is 8 coordinated. And for the calcium complex, as to the magnesium complex, they have the very similar structure like the marble flow. And then it's also the regular coordination structure of all of the transitional metal complexes. And I should to say that all of these metal complexes from start of the reaction in the lab, I told my students, you need to be notice two rules. The first is that you can only use water and ethanol as the solvent because you have to look forward to the future industrial production. So no other pollution should be here. So all of the synthesis of our complexes were formed in the water and ethanol solution. This is one. And the other, I told them, the synthetic temperature should be not too high. So most of the complexes were reacted about 50 to 60 degrees, not higher. So this is also considering the future enlargement. Okay. And now we primarily focus on the water solubility of the calcium complex of enrofloxacin. And this is obviously to solve the problem of the water insoluble of the enrofloxacin. And now from the detailed experiment, we can enhance the water solubility to about 0.4 gram. So that means we have enhanced the water solubility about 3 times than the enrofloxacin. So the coordination of the enrofloxacin to the metal ions won't decrease it. Okay, then we also screen the MIC and MBC for the metal complexes of enrofloxacin. And the results also showed very similar or very the same results with that of the marble floxacin. That means no obvious difference, only few of them showed some improvement, but we can basically say the anti-tumor, the antibacterial activity of the metal complex of enrofloxacin retained those of the ligand. So the coordination won't decrease or eliminate the activity, but retain. Okay, then we also test their acute toxicity on mice. And here we need to make a note that the sodium of the enrofloxacin. That means due to the poor water solubility of enrofloxacin, and when it was used in clinical, it has to be dissolved by some base, like the sodium carbonate. So actually, it was used as a sodium salt. So when we test the neutralized enrofloxacin, the LD50 value is about 5,000 grams per kilogram. So it can be classified into the grade 2. And as reported, it was classified into grade 3, but the LD value we achieved is just on the edge. So it's some close to that. But when we change it to a sodium salt, its LD50 values decreased dramatically. That means the sodium salt of enrofloxacin was much higher than its neutralized state. So that means the actual used drug of enrofloxacin are much more toxic, just as reported. So we use this experiment to make sure and understand why that was said. Okay, then our coordinated complex, like the calcium or the copper or the zinc complex of the enrofloxacin, their LD50 value are much more lower than the sodium one, also lower than the neutralized enrofloxacin. And so we firstly reported the copper one, and now the calcium complex was just to be submitted. And another metal complex interesting is the zinc. The zinc complex just showed like the lutetium complex of marbofloxacin, as we previously shown. So it is a non-toxic complex. So this is what we need to further explore and to say what was the inner mechanism of the zinc metal complex that much almost decrease or eliminate the acute toxicity. It was found that both metal complexes showed faster time to peak and higher plasma concentration in rats than the enrofloxacin. So they served the same retention time, but the faster time to peak and higher concentration. That means they showed the other wanted in clinic. And then this is the outline of the PK/PD properties, and we also notice that the area and the curve of the enrofloxacin calcium complex was more than 2 times of the enrofloxacin. So that means it will significantly increase the total amount of the drug into the blood system. And we also notice that the half-life of elimination of the calcium complex was about 4.4 hours. That is also much longer or larger than that of the enrofloxacin. That means it can serve as a long-term and more effective in higher concentration in rats. And on the other hand, we also learned that the enrofloxacin was not only used as a veterinary drug on the living stock on the land, but also popularly used in the aquatic products. That means for fish, shrimp, like that. So we also built the zebrafish platform to test the toxicity of the enrofloxacin calcium complex to make sure if it is also not so toxic to the aquatic products. Okay, this is the first 5 days 5 days duration of the incubation of our compounds with the zebrafish. And from the results, we can see that the calcium complex did not show higher toxicity on zebrafish comparing with the enrofloxacin, especially at a very high concentration of 20 μm, and it is much lower than that of enrofloxacin. And this is under the stereo microscope, we can see the whole body of the zebrafish, and to see that actually both the calcium complex and the enrofloxacin do not show the much higher toxicity on the zebrafish. And at a higher concentration, we can see the zebrafish can grow more normally as a control. And especially we can see from the liver and from the body length. And comparatively, if at the same concentration of the enrofloxacin, and the growth was slightly hindered, and so the body length was shorter. So the calcium complex also showed some lower toxicity than the enrofloxacin on zebrafish. And we also want to learn about what the possible mechanism for this kind of drug in the aquatic products, and to make bacterial activity. So generally, the bacterial will cause the inflammation effects in the animals, and so we build different kind of platform to research if our products or our compounds can reduce this phenomenon. And the first model was built by the copper sulfate, and it will cause the significant inflammation in zebrafish, and will give the significant green fluorescence in it. And then you can see with the control, the green fluorescence enhanced under the induction of the copper salt. And then we incubate our compound with the zebrafish induced. And then we can see in different concentrations of incubation, they can all effectively decrease the inflammation and status of the zebrafish. And comparatively, we can find that the calcium complex of enrofloxacin, especially in the under the medium and high concentration, it can effectively reduce the green emission of the fluorescence. So we think that the anti-inflammation effects of the calcium complex is also better than the enrofloxacin. The small model produced by the doctor that is a very strong emission. And then after the incubation with the complex or the ligands, and the emission was effectively reduced, and even at a lower concentration of the enrofloxacin and its calcium complex. So comparatively, you can see the metal complexes seems also more effective than the ligands to eliminate the ROS product, the ROS produced. Okay, finally, we use our calcium complex of the enrofloxacin to get a quasi-clinical trial on the E. coli infected broilers. And from the pictures, maybe some uncomfortable, sorry for that. And we can see the chickens, the healthy chickens give the liquid and sometimes with blood.
Dr. Liu, we cannot hear you. Can you unmute your mic?
Sorry, it's okay now?
Yes.
Oh, okay. Sorry. And then you can see that this is the main syndrome of the infected chicken or broiler. And then we use our calcium complex together with enrofloxacin to treat them respectively. And let's move to the table. And this is the result. You can see we choose the two types of broilers. One is AA type. That is much more heard in China. And another is the 817 hybrid broiler. That is a kind of broiler that has higher anti-infection. So we use the two types for testing. And now you can see the result that in each kind of product, we got the best result, both for the enrofloxacin calcium complex. And that means regarding to the survival rate, or that means the curative rate. No need for high doses of the complex, only the medium or the lower concentration is enough. And we also consider that the much higher concentration may be to do some side effects to the broiler maybe to broke the bacteria inside and leak out more toxins. So no need for the higher. And this is also including the average weight gain. And we can see that for two different types of bolus, the result is not the same. For the AA bolus, you can see the better to get the relative weight gain is for enrofloxacin, the ligand. Yes. So it gains 52% compared to the control. You should know that this is under the E. coli infection. So the growing, of course, should be lower than the control group. But for the 817 hybrid bolus, you can see at the lower dose of the calcium complex of enrofloxacin, it has a fast relative weight gain and is much higher than the other group. It reached the 47% of the control group. So when toward this, we get a much more positive result. But since the two types are not the same, so that means we need to proceed more clinical trials on different farms, and this is what we want to do next. And we think more results achieved and the clearer conclusion we can make. Okay, so this is almost a summary. Let's skip it because I have talked it. Okay, and also we get some research outcomes. This is what I want to say, that if we focus on the anti-tumor activities, studies of some metal complexes may be more easy to publish the papers. And for this, you focus on the veterinary drugs is not so easy. So we have to make an awkward balance to hold them both. Okay, we have two authorized patents for marbofloxacin, and the enrofloxacin, especially its calcium complex, is still suspending. We have applied for more than 3 years, but we have to defense again and again, because when you applied for the calcium of marbofloxacin, then the similar complex of enrofloxacin will be challenging. And so we want to prove it to innovative enough from other aspects, just like the synthetic. Okay, so finally, we think the most promising veterinary drugs should be focused on the calcium complex. This is mainly because we do not need to worry about the metal ions residue in the animals. This is sometimes was discriminated by most of the pharmacologists. So for chemists, we need to care about how to choose a proper metal ion. Okay, so I think my prospect is that we have to notice that the chemical drugs as a veterinary drug is more and more challenging. Although it still now cannot be replaced totally, but its window will getting more and more and smaller. And so I suggest that the Chinese veterinary drug is a promising direction. That means we need to get the lessons and experience both from the Chinese medicine research for the human beings and timely to convert it or to reference it to the development of Chinese veterinary drugs. And this is easy to say, but hard to do. So as previously reported, we also suggested that the active ingredients of the TCMs and should be a promising ligand for us to make the metal complexes, and their studies should not be limited to for human beings, but also for animal drugs. This is my suggestion. And I would also thank my dear collaborators and my hardworking students. Only not so many students, but they have do many works, not only in our lab, but also in many friendly institutes. I thank all of them, and also thank for the financial supporting. Well, this is my speech, and I hope there will be some helpful for you. Also, thank you for your attention. Welcome to the beautiful city of Guilin. Thank you all. Okay.
Very much, Professor Cheng. With this, it was a very nice and informative presentation and very effectively explained as well. If you allow me, I can take a brief question just to keep up with the time. So the question is why the natural drugs from plants are more effective than the synthetic drugs?
Sorry, would you like to speak slower? Yeah, sorry.
Okay. Why the natural drugs from plants are more effective than the synthetic drugs?
Okay, I think I cannot to guarantee that it is more effective, but I think the plants, the herbs were grown from the earth, so they should have the more much higher biocompatibility to the earth. That means the herb from the earth, and finally it will die and go back to the earth. And so as the main ingredients, that means the main products from the herbs should be more biocompatible to the earth. So we and the animals all belong to the earth. So I think it will be more friendly to us. So this is suggesting will be more biosafety. Can you understand me?
Yes, yes. Thank you very much.
Okay,
Professor
thank you. Thank you.
With this, I would request Professor Dr. Iqbal Chaudhary to kindly say a few words for the whole presentation of Professor Liu.
Professor Liu was an excellent presentation, and you covered a very important aspect of medicinal chemistry, which is veterinary sciences. Also, your comments about the linkages between the health of animals and human being are extremely important, because this whole concept of one health is very, very important. We all understand that, yeah, exactly, these pandemics are all zoonotic, and it is important to have healthy environment, healthy animals, and only then humanity would also be healthy. So thank you very much for delivering an excellent presentation and excellent display of a high quality chemistry. Thank you.
Thank you, Professor. Thank you.
Thank you.
Thank you very much. And thank you to Professor Liu as well. With this, I would like our participants to know that we come to the end of the first technical session of our second day of the online meeting. We will begin our second session of the online meeting at 1300 hours Pakistan Standard Time. And we have some of the very fascinating and interesting talks coming up in our next session. So we thank you for joining us today in the first session, and hope to see you all in the second session at 1300 hours Pakistan Standard Time. This is for the lunch and the prayer break for all participants. Thank you. Welcome back. Welcome back. And with this, we will begin our last technical session of the two-day meeting on the recent developments in the field of drug discovery and medicinal chemistry. And with this, we have our next speaker is Professor Dr. Mohammad Saeed. Professor Dr. Saeed is from Lahore University of Management Sciences. The title of his talk today is Targeting Viral Proteases for Designing and Discovering Direct Acting Antivirals. And with this, I would request Dr. Hina Siddiqui to kindly introduce our guest speaker, Dr. Saeed, to the participant.
Sure, Hazima. Assalamu alaikum, everyone. Good day. It's my pleasure once again to introduce a very eminent speaker, Dr. Mohammad Saeed, who is associated with LUMS, which is one of the prestigious institutions in Pakistan. Dr. Saeed has joined LUMS in 2014 as an associate professor. He established the Bioorganic and Medicinal Chemistry group in Department of Chemistry and Chemical Engineering. He is originally trained as a synthetic organic chemist, and he has very extensive postdoctoral research experience at the interface of chemistry and biology. He is entrusted to address the modern challenges in biological sciences and medicinal chemistry by using tools of organic chemistry. More specifically, he pursues rational antiviral and anti-cancer drug designing. Dr. Saeed has received his PhD degree in organic chemistry from University of Tübingen, Germany, under the supervision of Professor Wolfgang Folter. And during his studies, he has synthesized several natural product compounds through total synthesis approaches. And after PhD, he joined the research group of Professor Iqbal at the University of Nebraska, USA, where he participated as a chemical biologist in mechanistic investigation of cancer initiation by estrogen and related carcinogenic compounds. And before joining LUMS, he has also worked at University of Iowa as the visiting assistant professor and as a postdoctoral fellow also. He has published around 50 research articles in reputed international journals. He is currently investigating proteases of different viruses, COVID-19, dengue, HCV, as drug target for designing efficient direct-acting antiviral. With this introduction, I request once again Professor Dr. Mohammad Saeed for his scientific lecture.
Thank you very much, Dr. Hina Siddiqui, once again for the introduction and invitation to present my research that I am currently doing at Lahore University of Management Sciences. As you know,
sir, we cannot see you.
Let me see. Hold on. Yeah,
Now it's okay.
Yeah, my video is active from my end. I think it should be visible.
Yeah, it's okay now.
Okay. So actually, before joining LUMS, I spent several years, investigating the mechanism of cancer initiation, which in more specifically, you can say it's the tumorigenesis area, where we have used some estrogens as a model to induce mutation, cancer-specific mutation in breast cancer and prostate cancer. However, after 2014, I have to shift my direction little bit toward the current areas, current challenges in Pakistan. So when I came to Pakistan, then there was frequent outbreaks of dengue virus during that time. So instead of following the direction of my postdoctoral research or my PhD research, I just tried to establish a facility at LUMS, which is called the Bioorganic and Medicinal Chemistry, which is intended to deal not only toward understanding the mechanisms of disease initiation, but also provide some solution to chemically intervene some biological pathways and propose some structure which can be developed further into potential drugs. So in this presentation, I will present 4 different areas in which I have worked up to now. And we have published around 10 publications out of this work. Let's start. So, okay, so everybody knows that the Pakistan is under a continuous threat of viral infection outbreaks. For instance, Pakistan is the second in the world where the hepatitis C virus have the is the heavy burden. Although this disease is now under control, and we are expecting to finish this virus by 2030 for most of the countries in the world. Okay, so in 2011, for example, the dengue infection in Pakistan that made the global headings, raising the concern all over the world. And with the with some programs which are run by government and by the extensive usage of fumigation techniques, we don't have that much severity of this disease, but occasional outbreaks are still occurring in Pakistan. And this dengue virus actually contain 4 different serotypes. And that is the big problem, that almost all of these 4 isotypes, they are prevalent in Pakistan. And if someone is infected with the serotype 1, then it is self-limited disease, and most of the time person recovered from this infection and they develop antibodies. The problem happens when a second isotype, like dengue 2 or dengue 3, they infect the same person who has already antibodies or developed the immunology against dengue 1. Then this infection is not limited to the minor disease. It has developed to more fatal forms of diseases, which are called the dengue shock syndrome or the dengue hemorrhagic fever, DHF. And that is the biggest issue, that when several serotypes of these viruses, which are, by the way, from the flavivirus, flaviviridae family, if they infect, then they increase the chances of fatal infection by a mechanism which is called the antibody-based enhancement, or ADE, antibody-dependent enhancement. And from 2018, another flavivirus called the Zika virus has also, come into the game. And due to international travels, there are chances that the people who are already infected with any one serotypes, and if they are infected by any other serotype of dengue or Zika virus, they are under threat of developing this fatal form of dengue fevers. Okay, so what are the current management or the clinical management of dengue infections? First of all, there is no direct medicine available to treat the dengue infection. Sanofi Pasteur has recently launched a vaccine, which they claim that this is a pan-specific, meaning that it covers all the serotypes of dengue virus. But there are several issues or questions which have been raised in different places of the world. For example, in a couple of years back, there were 3 deaths in children in Philippines who were vaccinated with dengue. And it was concerned that these deaths are associated with that vaccine. So it is still questionable whether people should be vaccinated using this therapeutics. So in Pakistan, how we are dealing dengue virus infection? We are actually trying to control the spread of Aedes aegypti mosquitoes, which are the vector of this virus. And by limiting the population of these mosquitoes, it is thought that we will not be infected. But this technique or this strategy involved a lot of fumigations with insecticides, and that is not eco-friendly and environmental friendly treatment. And it is claimed from different labs that some mosquitoes are developing resistance against the insecticides which are used in these fumigations. Therefore, there is a big demand of developing direct-acting antiviral drug against dengue, along with the effort to improve the vaccination strategy. So how direct-acting antiviral can be developed against flavivirus or other type of RNA-based viruses? So anyone who is interested to initiate work in this area, then I will request to pay attention in this slide, because that describes the whole strategy of drug development in this direct-acting antiviral area. So dengue virus or any flavivirus that is transmitted to human by the bite of mosquito, Aedes aegypti mosquito. And once it is in the skin, in the tissue, then it is endocytosed by the host cell. There are several dendritic cells or other immunology-based cells which serve as the host of this virus. So this virus is actually endocytosed by the cell, and once it is inside the cell, then endosome is opened, and RNA genome is available in the cytoplasm. And from here on, all RNA-based viruses follow more or less the same strategy. that this RNA is immediately translated by using the ribosomal machinery of the host cell at the location of endoplasmic reticulum. And depending upon how many open reading frame are available in the genome, the RNA is translated into a long peptide, which is called the viral polyprotein. And many structural and non-structural proteins are still attached in this long thread. And if the process is stopped at this stage by any way, then that is the end of story as far as the virus is concerned, because these components, they are useless at this stage. Therefore, the virus is actually clever, and it uses some protease of the host cell as well as its own protease to cleave this this polyprotein. This cleavage is required for the release of this protein and the post-translational modifications. For example, this protein NS5, that is the RNA-dependent RNA polymerase, and that is used to make the additional copies of RNA virus. If it is attached in this form, this is a useless piece of peptide. And another protein, which is again exposed in the cytoplasmic site of this endoplasmic reticulum, that is called NS3. It has the proteolytic activity. It can cleave not only itself, but it can cleave the other loop which are exposed in the cytoplasm site of the ER. And now the loop which are located inside the lumen of ER, they are processed by the host proteases like signalase and furin. And these component, mostly they are the the component or the structural component of virus. For example, we have the PRM, precursor membrane enveloped protein that is also located in the lumen, and these are processed by the host proteases. With the action of all these proteases, this polyprotein is processed further into the functional structural and non-structural protein. And the most important one, the non-structural protein, because they have the enzymatic activities that they are used to make the RNA copy at one end, and also when the structural proteins are also synthesized at the same time, then the RNA and the structural part of virus, they are assembled together to make the active virions. And then they will continue the infection in the new host cells, and then the whole cycle will be repeated again. So our hypothesis is that if we stop the activity of viral proteases, then we will not have the functional NS5 protein, which is the RNA polymerase. We will not have the capsid, which is also located in the cytoplasmic site. So when these important proteins are not available, then the assembly of virus will be affected, and we hypothesize that after this inhibition, the whole cell cycle, the viral reproductive cycle will stop, and infection can be inhibited. Okay, so here is one validation of using protease as the drug target, and I have presented in my last presentation yesterday by presenting the example of HIV, that HIV was the first infection of HIV was reported in 1981 in America. And then within a few years, like 3 or 4 years, I think in 1985, they developed the first inhibitor of the reverse transcriptase, which is called the azidothiamidine AZT. With only single therapy or monotherapy of azidothiamidine, the deaths were not stopped significantly. And in 1995, they developed protease inhibitor sequenavir. And when these two drugs were used together, then the death rate was declined significantly. And now we have several protease inhibitor which has been approved from FDA, not only for the HIV infection, but also for HCV infection. And we hope that by using the protease of dengue virus and even SARS-CoV-2, there are potential to develop efficient drugs against these diseases. Okay, so the dengue virus protease is actually a complex of two non-structural proteins, namely non-structural protein 3, NS3, and NS2B. So NS3 is a catalytic domain, and it contains catalytic triad, which is based on the serine, histidine, and aspartic acid. When there is no cofactor attached to NS3, then it is very weakly active. And the reason is that the protein itself is loosely folded, and the catalytic triad is not properly oriented, and the reaction is very sluggish to cleave the protein. But as soon as the cofactor is available, then cofactor will wound around the NS3 domain, and it will not only bring the catalytic triad in the proper orientation, but also if you see the C terminal of the cofactor, then it is actually induced to a beta hairpin, which contribute toward the substrate binding site. So the substrate binding site is just right here where you are directly looking at. And the different subsites are identified. Then subsite 2 and subsite 3, they require the contribution from the cofactor for the fully development. So with these two proteins in the complex form, the virus is very efficient to cleave its polyprotein, and it helps in the assembly of the virions. So our working hypothesis is that any natural product or synthetic compound which can inhibit the function of this complex can stop the virus replication cycle, and the compound can be developed further into direct acting anti-dengue agent. Okay, so in my lab, I have developed a biochemical assay by using a tetrapeptide substrate, which is commercially available, and this substrate is attached with the fluorogenic moiety, which is a chromine. And when it is attached at the peptide, then it does not fluoresce much. So the fluorescence is quenched. So this kind of strategy is named as FRET-based substrate in the literature, and it's a well-known methodology to develop the biochemical assays. So the NS2B, NS3 complex that was recombinantly expressed in the bacterial cells and having these two things ready, then we were ready to screen different libraries of compounds and different synthetic compounds. So the the mechanism of the biochemical assay is that the catalytic triad, they attack at this amide bond, and it is cleaved at this position, and then coumarin is freed or released, which fluoresces when you excite it at wavelength, at a different excitation wavelength. So having the biochemical assay in hand, we embarked on the screenings, different plant extract or herb extract. So we have selected some herbs which are commercially available, and our objective was to screen those natural product, those plant or herb extract which have known history of utilization in the febrile diseases in alternative medicine. Okay, so these these plant extract or plant material, they were extracted in ethanol, and with a single dose of 200 microgram per ml extract, we have tested our biochemical assays, and we found that S. aromaticum, which is the clove, by the way. So the extract prepared from clove, they have almost identical inhibition as given by the positive control of A protein. So we screen these plant extracts at different doses to find the dose response and IC50 in the extract level. And again, the clove, they have the efficient IC50 against the dengue proteases of serotype 2 and serotype 3. So these are two serotypes which are prevalent in Pakistan. So we have, just focus on these two proteases in our study. Okay, so after identifying the plant extract, we conducted bioassay directed isolation and identification of the phytochemical which is responsible for this activity. So we have first fractionated this ethanol extract, and each fraction was tested against the dengue virus protease, and unfortunately, or maybe fortunately, we just find the activity in the aqueous fraction, not the conventional fractions like chloroform or the slightly less polar, because clove is mostly famous for the non-polar fractions. But here we have just find that the aqueous fraction is giving the highest activity. So we use the semi-preparative HPLC to blindly collect different fractions, like 9 HPLC eluent were collected without any resolution of the peak, because the extract is very complex at this stage. And then we have conducted the protease assays again, and then we found the fraction which is eluted at approximately 40% methanol in water that have the highest activity. And this eluent was repeatedly purified on a semi-preparative column. And at end, we identified 3 compound natural product, isobiflorine, biflorine, and eugenine as the most active compounds of this fraction. So here is the drug dose response by using the purified component, purified natural product, and eugenine is way much active then the bifluorine and fluorine component. So we focus our attention on the eugenine for the further study. So here is the quantitative data of IC50. Serotype 2 was the most effective, and eugenine, if you look at that, this is a very nice activity in the nanomolar scale, which is also effective in the serotype 3 as well. Okay, then we went to find the bond atomic level interaction of protein and the isolated compound by using saturated transfer difference NMR study, which is a well-known experiment. So here, if you see, the red is the NMR of the natural product, the purified compound, which have different aromatic peaks, which are labeled A, B, C, and some peaks in the carbohydrate section in the sugar moiety. And the blue one is the NOE difference spectrum. So this technique tells that if there is interaction between ligand and protein, then the magnetization is transferred and transferred to the ligand, and based on the percentage enhancement of the signal, you can find out the interaction pattern. So which side is interacting with the protein. So this experiment tells us that the aromatic region, that is the mostly interacted with the protein structure, along with some peak of the carbohydrate like H1 at the proton, at the anomeric proton, that has 55% enhancement, and very small enhancement from the H2 and H3 protons. The similar data was also found for isoflurane and the bifluorine and isoflurane, and we again found that the aromatic region is the one which is demonstrating this interaction through some sort of hydrogen bonding with the residue in the catalytic site. Okay, then the mechanism of inhibition was investigated when we have obtained the Lineweaver work plot between the substrate concentration and the rate of reaction. And the plot indicated that this is some competitive type of inhibition, which means that the natural product, they bind at the catalytic site of enzyme, and which was further confirmed by the docking analysis. And if you look at the this protein, that this has two different colors. So gray color is for the NS3 component, the catalytic site, and this blue is the cofactor side chain or beta hairpin, which has contributed toward the development of this catalytic site. So the biphenyl moiety of eugenine that sit nicely in the subsite S1, whereas the three gallic acid moieties, they are also oriented in the S2 site here, and then S3 and S4. I'm sorry, I forgot to label these sites, but this is S1 site, subsite, this is S2 subsite, this is S3 subsite, and then we have the fourth subsite, S4. And we can identify through this docking analysis different residue which are involved in the hydrogen bonding, and the van der Waals interactions with the different residues. So identical data was obtained for the isobiflurine, which bind at the S2 subsite, and S1 site is empty here. And if you recall my yesterday's talk on the drug designing techniques, then this moiety can be used as a small fragment which bind at the S2 site, and you will see that this moiety, this molecule bind at the S4 subsite. And if you can combine these two molecules, we will have very efficient fragment-based lead from this study. And the further work is still going on in this direction, and this initial results have already been published in ACS Omega. Okay, then the second project which I want to present is the screening of commercial library. So we have purchased a library of inhibitors which have activity against different enzymes which are overexpressed in cancers and other diseases. So we can call to some extent this is the drug repurposing effort. So we use this library to screen against the biochemical assay. And here in y-axis, we can see the percentage inhibition, and on x-axis, we have all compounds located. And we have selected the top 21 compound which have shown inhibition greater than 90%. And all other compound we did not screen, and we drop from the further study. So here is the flow sheet that how this study went further. So at 25 micromolar dose of the inhibitor, we screen our assay, and from there we have identified 21 molecules which have shown more than 90% inhibition. And further filtering through different artifacts like false positive or inner filter effect, then most of these compounds were removed because they were either showing activity due to aggregation or due to quenching of the fluorescence from the probe. After removal that, we have identified 3 inhibitors which are interestingly the inhibitor of BCL2, which is the oncogene in several type of cancers. In this slide, I will just briefly overview for the further investigation on these compounds. So 8101 is a BH3 mimetic inhibitor of BCLX and BCL2, and it is in clinical phase 2 in a clinical trial. And these clinical trials are, by the way, in the cancer area. It has nothing to do with the viral side. So it's the mechanistic investigation tell us that this molecule have a mixed type inhibition with IC50 of 910 nanomolar and Ki in the range of sub micromolar concentration. TW31 is a non-peptide inhibitor of BCL2 and family of these kinases. In our study, this was found to inhibit dengue protease serotype 2 at IC50 of 1.66 micromolar. With the single dose of 25 micromolar, it inhibits this the dengue protease in 100%. And the mode of inhibition turned out to be a competitive type with Ki in 1.52 micromolar range. And the third compound is again a competitive type inhibitor with the IC50 value of 3.16, and originally this compound is used as an inhibitor of BCL-XL family of protein. So this is the docking analysis, which tell us that the binding of ABT 337, it binds the dengue protease at the almost all subsite of catalyte binding site. It showed the hydrogen bonding interaction with residue of a cofactor and the residue of the domain factor, and several nonpolar interactions in different areas of NS3 protein. Okay, so the third project is actually our collaborative efforts. So one of my collaborators from Dr. Bilal Khan from University of Azad Kashmir, he sent us some compounds which he have developed through this reaction scheme. And he has developed 4 different libraries of oxadiazole and sulfonamide hybrids, where the structural variations are shown. So in the first 2 libraries, this moiety was constant by varying the thymidine or thiobenzyl moieties, and in the lower one, the scaffold was slightly different. So instead of nitrogen in the left side, the nitrogen is in right side in the bottom libraries. Screening of these compounds with the single dose of 50 micromolar, we found that the library A is most effective, which have shown the activity in the 90% range. So we have not conducted any study in the remaining molecules, but we have find the important lead from the library A. This showed the dose response, and from this dose response, we identify the molecule 8G and 8H as the potential hit. We have seen 8D also in the same range, but if we look at the the previous slide, then yeah, here. So the 8D, if you look at, then it has several methylene which are present between S and NH2. So that has introduced a lot of rotatable bonds. And according to Lipinski rule, if you have more than 10 rotatable bond in a molecule, then this is not a good hit. So you have to drop that. And using that reasoning, we have dropped the 8D from our study. So 8D was 8G and 8H. They were tested in dose response experiment, and this graph showed the line Weaver Burke plot of this compound, and IC50 turned out to be in a decent range. And here is the docking study, which tell us the atomic level interaction or map of the interaction We have several type of nonpolar interactions and hydrogen bonding with the important residue of the protease. Okay, lastly, I will present a very small fragment-based library. We developed a very small, fragment, which is called the benzoisothiazolinone, which is commonly called as bits. So these bits were synthesized in a single step reaction, which we have reported recently. By using this, reaction, we developed 15 different compounds, 15 or 16, and their screening with the single dose, give us information that the compound which have the aromatic moiety at the end position, this nitrogen, they are mostly active than the aliphatic members. Dose response also confirm this finding, and the best three components, which are the 5J, 5M, and 5N, these compounds were further tested, and their IC50s were in the sub-micromolar range, and this result have already been published, as I mentioned before. So I will summarize my talk by just pointing, highlighting the main finding of our studies. So our study tell us that the clove extract have a very potent ingredient which have potential to be used as a antiviral drug against dengue treatment. And eugenine and isobiflurine and biflurine, these are the active ingredient in this compound. And since this is proposed that these compounds can be repurposed toward the dengue treatment. By screening the oxadiazole and sulfonamide libraries, we identified two compounds which which are potent allosteric inhibitor of dengue virus. And similarly, the BIT inhibitor or fragments, they have been identified, and the three fragments are the potent allosteric inhibitor, which can be further developed into the potential lead. Okay, so in the end, I would acknowledge some of my research team members and some collaborators and some financial support from two sources. So from my own lab, the most of the work, the bioassays were conducted by Nosheen Slim, and she's currently a PhD student at the University of Göttingen. Farwa Batul is my PhD student, and she is writing her PhD thesis right now. Binish and Hafiz Javed, they were the MS student who have contributed toward the synthetic work and some part of the isolation. So I would like to thank Dr. Shahzadul Hasan from Biology Department and Amir Faisal to collaborate with some techniques like NMR study and sharing the cancer libraries with me, and Dr. Bilal for sharing his compounds under collaboration. And by the way, I'm open for any person who just who want to get their compounds screened by our biochemical assays in future. Financial support from the internal funding from LUMS and external funding from HEC is highly acknowledged. And I thank you again, the organizers of this wonderful event, and Dr. Hina Siddiqui, Khazima, and Professor Iqbal Chaudhary for giving me this opportunity to present my work. Thank you very much, and I'm open to answer any question.
Thank you very much, Professor Dr. Saeed. It was indeed a very refreshing as well as informative presentation. And with this, if you permit me, I would like to take a few questions for you.
Sure, yeah, please.
So, so one question is from Mr. Hassan. He's asking, keeping in mind the ADE mechanism, a type of ERD, by antibody-based vaccination and drug therapy, should common population be forced for taking doubtful vaccination if already have acquired national immunity? I mean, I don't know if that is directly related to your presentation or not.
Well, I think the dengue vaccine, I'm not qualified to answer this question, but I can just give you some information that many countries are allowing their population to be vaccinated. The population which is above 18-year-old, and most of the side effects which have been identified, they are found in the children, especially the population which has never been exposed against any flaviviruses before. So they somehow develop not the antibody against all serotypes of flaviviruses. Instead, some of antibodies against some of the serotypes are better developed than the other one. In elder population, I did not read any paper, any information or any news where very fatal side effect has been shown. I think that's it, what I can say about the vaccination using dengue vaccine.
Thank you, Dr. Saeed. I think there is also another question that is somewhat related to vaccines, but I think in connection with dengue virus. Let me read it out to you. Dear Dr. Professor Saeed, the COVID-19 vaccines are mostly mRNA, while the COVID-19 Delta virus is also infecting the vaccinated persons too. Do you think that RNA or mRNA-based vaccines are feasible to get protection from COVID-19, as you described in dengue virus proteases reactions? I think followed by another question. Which plant extracts you suggest to control COVID-19 infection?
Well, I think I will first answer the second question because that is more relevant to my area. So the COVID-19 is a newer disease, and although many people have previously worked in the earlier form of this virus, which are SARS and MERS, and they have reported several compounds from natural product which inhibit the earlier version of this virus. As far as the new virus is confirmed, I think I think in the previous year, most of the lab, they remained closed, I think all over the world. And mostly people have screened different libraries in a virtual way by the computational modeling. And we have, ourselves published one paper in the dengue virus where we, in the SARS-CoV virus, where we reported that some nature product like flavonoids and their synthetic analog containing sulfur in place of oxygen. They are very potent inhibitors, in vitro inhibitor of SARS-CoV-2 protease. The experimental data is still, not available. I am not aware of, at least at this point. But many people would like to screen different nature products to test whether any natural product have potential against the COVID-19. So as far as my lab is concerned, we are completing our recombinant expression of COVID-19 proteases, and I think within a month we will be ready to screen different libraries and natural product using the biochemical assay of SARS-CoV-2.
Thank you very much, Professor Saeed. And again, I would like to thank you for taking out your valuable time and be part of this online meeting. And with this, we will move on to our next speaker, Dr. Riazuddin. Dr. Riazuddin is from International Center for Chemical and Biological Sciences, University of Karachi. The title of his talk today is Computational Tools and Techniques for Early Phase of Drug Discovery Research. And with this, I would request Dr. Hina Siddiqui to introduce our next speaker, Dr. Riazuddin.
Thank you so much, Khazima. And it is such a pleasure to introduce my colleague, my friend, Dr. Riazuddin, who is Associate Professor at the Dr. Panjwani Center for Molecular Medicine and Drug Research, International Center for Chemical and Biological Sciences, University of Karachi. Dr. Riazuddin is currently working as an Associate Professor at PCMD, University of Karachi. His focus area of research is computational biology and bioinformatics. The research is focused on the application of new computational methods in order to solve medicinal chemistry related problems such as drug discovery against infectious diseases. His particular focus is the resistant microbial pathogens, for which the enormous amount of genomics data is available to be analyzed by the latest technologies introduced in the areas of computational biology and bioinformatics. Dr. Riaz has 55 peer-reviewed international journals, which he has published in High Impact Factor International Journal. In addition to this, Dr. Riaz has secured lots of competitive international and national funding to establish his laboratory. And with this, I request Dr. Riazuddin for his talk.
Assalamu alaikum. And I just want to share my screen first and then I will start. Share screen and then this option. That is okay. Sure. All right. I hope everyone can see my screen. Khazima, can you confirm?
Yes, Dr. Saeed. Sorry, Dr. Riaz, I can see your screen.
Okay, so Thank you so much, Dr. Hina, for introducing me. It's a great honor to be participating in this prestigious online talk, which Comstech has arranged. And the distinguished speakers I have been listening for the last 2 days actually is amazing. And my topic today is to mostly talking about these computational methods and tools which you can use for the early phase of drug discovery research. Because my focus of research is more like structural bioinformatics as well as chemoinformatics. So, and I have earlier background of chemistry, so I would like to blend all of this together. And as I can see lots of students in the participation actually. So I would be starting with brief introduction with the topic, and then moving on further with my research, which I have been doing for last couple of years in this institute, and I will also share some literature examples as well. So with that, moving on further to my first slide. I hope everyone actually, as long as you are human, you might have encountered cold at some point of your life actually. And that cold is actually giving you so much symptoms of flu and colds like fever, headache, and others, you know. So have you ever wondered, and sometimes, you know, you have got maybe food poison, you know, so, and measles or chicken pox, you know. Have you ever wondered like what exactly caused that disease? What exactly caused this kind of things and disturbance in your body? It's basically, I would say, it's basically microbiology or micro world actually, which actually causing and such kind of diseases. So for that, we normally go for like whenever we have fever and other kind of symptoms, which actually give you a sign like you are having infection, so you get antibiotics, you know. But mind it, like for virus, it does not work. So microbiome is a real world. It's parallelly existing around us, you know, and that world is basically, you know, like giving some tough time to the humans actually. And they are everywhere. They are on your skin. They are around the soil, maybe in the lawn, in park, everywhere. The microbes are there, in your mouth, in doorknob, you know, and everywhere. So, but don't worry. Like every microorganism is not pathogen. The pathogen is the microbes which are pathogen. They are the one actually who actually got trouble. So humans are having always encounter in the history. If you talk about the real plague happening in 1630, for example, the real pandemic happened back in the 1600. So you can imagine like such kind of disease has always been present in human history. And this is possible only late 19th century when scientists were able to discover some methods and some techniques or some kind of, like I would say, tools which can help them to see that microbial world. So famous historical actually discovery made by Hans and Zacharias Janssen back in 1595, where they have made a very simple microscope with having only a tube with 2 lenses into it, which can give you which can give you a look of about 9 to 10 times higher resolution about or magnification of the microorganism. So it's a big breakthrough starting back in 16th century, where actually they have got this tool to be developed. And moving on further, about 100 years after, Anthony von Leeuwenhoek, actually he was a cloth merchant, and he was actually the one who actually has further advanced this microscope technology. And he was actually the one who first time saw some kind of living creatures in a drop of water, and that living creature, the microbes actually inside this water samples, and he named them as animalcules actually. And further later to further a century after, Friedrich was the one who is a German physician actually. He developed a germ theory of the disease. He said like whenever you have certain kind of diseases like measles and other kind of infectious diseases, you have a a bacteria or germ responsible for that. That developed the real theory of germs regarding the disease states. Moving on further, like Louis Pasteur and Robert Koch, you guys must have known their name. They are very famous, and they are the ones who actually demonstrated that the microorganism causing the real reason of the diseases such as cholera, plague, tuberculosis, and rabies. So that further advanced the theory regarding the disease state and its responsible organisms. So again, animalcules, or later on people named them as microbes, they are of every different shapes, you know, they are everywhere. For example, if you talk about bacteria, they are having different kind of arrangements and different kind of cellular life. And one thing is common in every microorganism, they are basically unicellulars. So this microorganism also include viruses. For example, this coronavirus, recent pandemic, is basically as a human being affected by coronavirus, and they are even smaller than the bacteria, and they can cause a disease to not only human but also the bacteria. So this is a big challenge. And as I said earlier, like some of the microorganisms are multicellular. They could be considered as like you know, prokaryotes, but they are very, very tiny. And unicellular organisms are the big class of microorganisms, and they include bacteria and others. So if you talk about unicellular, that means it contains only one cell. And what is the difference between prokaryotic and eukaryotic cells? Is basically the nucleus structure. You know, it does not have in prokaryotic cell, it does not have any kind of membrane around it. So that's why the nucleus of prokaryotic cell is normally dispersed in complete cytoplasm. So why we are talking about this cell actually? Cell is the living organism basic unit, and the nucleus of any cell is basically containing the chromosomes, and chromosome in chemical terminology, it's basically DNA, which is deoxyribonucleic acid. And with that, I would like to play this video here, and I hope everybody can see. And this video will more actually clarify regarding the cell and its structure. It will explain more about what I'm talking, you know, today. I hope it's played for everyone.
Yes, Dr. Riaz, it is being played at my end.
Sorry, say again.
Yes, it is working perfectly fine at my end.
Okay, thank you. Oops, sorry. Forward it. So this is helical DNA, and which has genes onto it, which are nothing but the sequence of nucleotides. I will later talk about this DNA structure discovery back in 1962. And chemically, these building blocks are nothing but the nitrogenous bases. 4 bases called adenine, thymine, guanine, and cytosine. All right, so this is another video which I would not play complete, but I will play a little part of it so that it could give more context about my talk today, and it has some voice also. so moving on further. So as I said, like this video has given me has given you all the context of this presentation, and so we were. You can imagine, like I'm going to talk about more DNA and structure, which is being described by Watson and Craig back in 1950s, where they have worked together. They have got Nobel Prize for that, and they have actually come up with the double helix structure and arrangement of the DNA. And that explains lots of genetics and genomics information, which actually we are getting benefits of now. So we are in the midst of a golden era of biology, where DNA has been, the structure of the DNA has been solved. Genomics is a big science these days, which gives you every organism's genomic structure and the sequence, and which you can use it for the purpose of drug discovery and drug designing. So why actually very important and significant to learn about DNA and its structure? So central dogma of molecular biology says that DNA translates its information transcribes its information to RNA, which in turn translates information into protein. And from coming from DNA to protein, like from genotype to phenotype, there are lots of level of information available. For example, you can have lots of sequence and structure information about the RNA structure and function, and later on protein sequence and its structure and function, and that gives rise to really the phenotype of this organism. So from DNA to protein, there are lots of information available to be explored for the purpose of drug discovery research, particularly in the early phase of drug discovery. So Human Genome Project, very famous. It was being sequenced in 2001 or 2, I guess, and it says like it is comprised of 3 billion bases, which covers about 30,000 human genes. It is quite low in number compared to Drosophila only. But because humans are more complex organism, multicellular, that's why they have multiple function of a single gene. So that's why low number of genes can still code lots of proteins. So I could talk a little bit about the historical movement or historical development of bioinformatics or structural bioinformatics. You see, in the first protein database was actually being developed by Margaret Hoff back in 1950s. And if you talk about comparison of the hardware available now and then, so for example, it's a very simple comparison between a computer found in 1960, which is, can you see this the size of the huge hardware in a big room requires to run a computer with a RAM of only 32 kilobytes, which is nothing, compared to these days, because our phone is even have more than that. And the processor of this machine, which is a big huge, is about only 2.18 megahertz, but the microhertz, but the cost is like $2.9 million, which is too much. And if you talk about in 2008, for example, a simple 1 GB RAM and 2.4 gigahertz processor computer from Apple was about $11.99. And if you can see recently, like this kind of specs and laptop available with very low price. So computational power actually, I would say, increased dramatically from the day one when the computer started. So that leads to use this computational power for every kind of scientific research, for example, biology and chemistry. So is it very important to know your personal DNA sequence? That's the question actually. Do you really want to? And if you want to know the DNA sequence, does it help to understand the disease mechanism? So this kind of question we can answer in our presentation today, and how to characterize new disease, and what new treatments can be discovered by using this information, and how do we treat individual patients. These are the personalized medicine, which is a big area of research these days around the world. So from now, the outline of a presentation is to talk about how you can use computational tools for the early phase of drug discovery research, which includes methods, for example, molecular docking, pharmacophore modeling, quantitative structural activity relationship, protein flexibility, for example, molecular dynamic simulations. And Mostly I would be, you see, I can remember the presentation given to this forum last day by Dr. Saeed. Actually, he talked about much of this molecular docking and other visual screening application in his presentation. But my focus today is to introduce you with this technique and how you can use it for early phase of drug discovery research. So moving on. So modern drug discovery, I would say it's a multidisciplinary area of research, which contains lots of things all together. It cannot be run alone. So you have to work together, you like combinatorial chemistry, bioinformatics, and computational medchem, molecular biology. Every kind of experts sit together and then solve a typical problem of human pathogenesis actually. So if we name that discipline in a very historical terminology called computer-aided drug design, so if I define it, actually it's a specialized discipline that uses computational methods to simulate drug receptor interactions. So how drug is going to the body and how it works there, that's basically simulate in computational environment. And those interactions which drug makes with the human body can be electrostatic and statics in nature due to its chemical nature. So what kind of normal topic you covered during any kind of course covering computer-aided drug design is basically molecular docking, as I said earlier, and pharmacophore modeling. So if time allows, I can give you a very quick one-line explanation of each topic. For example, molecular docking, which I will cover more in detail later on. But if I say slowly, or actually more quickly, docking is nothing but how two molecules interact in human body. You simulate into a computational environment in which one part is the ligand, small molecule, and the other is other is basically the macromolecule, which is most of the time is protein. And the pharmacophore modeling is not, as I remember again, Dr. Saeed has explained yesterday. And that is nothing but if you do not have any kind of macromolecular structure and you still have some active data, active compound data, you can model them by using their 3D structures and then find the structural requirement for the purpose of having this activity, which is being displayed by these compounds. MD simulation is one step forward of the docking, where actually in the docking you have a static system which is being moved into these MD simulations, and you actually allow protein as well as the ligand flexible inside the environment of this solvent, and then you understand and model the real dynamics of the human body and the macromolecules along with the ligand structure. And virtual screening is basically using structure-based methods or pharmacophore-based methods to perform on a large chemical space so that you can bring few predicted active compounds against certain drug targets. I will show an example of it from my work later on in this presentation. And QSAR is a very, very powerful technique because it allows you to find a model, mathematical model that explains the requirement of the structure for the purpose of displaying the activity of that particular class of compounds. And it has another level of, I would say, quality data coming up from QSAR. Actually, these days there are lots of different kind of QSAR, like 2D QSAR and 3D QSAR, in which 3D QSAR comes out to be very, very powerful in terms of prediction of unknown compound activity. So yeah, so this kind of topics you always have encountered in such kind of computer-aided drug design presentation or topic, So let's move back to molecular docking and its principle. So how drug acts? If you understand, if you remember your textbook, biochemistry textbook actually, and you recall that drugs work by binding with a macromolecule, and during that binding, they form certain kind of interactions, and those interactions are driven by the chemical nature of receptor as well as the ligand, which is binding towards this macromolecule. And those interactions are responsible for exhibiting the I would say binding affinity of that particular compound. The classical lock and key model between enzyme and its substrate is basically saying, like, if the compound or ligand is binding tightly, it reflects more binding affinity towards that particular enzyme. And if that enzyme is involved in certain kind of pathogenesis, then you may get advantage of switching off the activity of that enzyme by binding another ligand with it instead of receptor. And molecular docking, it basically uses that same principle, and but using computational tools or mathematical models. And the principle remains same. A small ligand is going to bind with a macromolecule inside its binding site, and then you actually estimate the binding energies or binding affinities, or I would say by using some mathematical modeling, and you can predict which compound is more tightly binding or which compound is going to be less likely binding to the same receptor. So molecular docking is basically consisting of two steps. One is finding the 3D orientation of that organic molecule inside the binding pocket. So that covers by the search algorithm. And after that, if you get the best possible energetically stable orientation or confirmation of that ligand, then you estimate the binding affinity by using mathematical equations called scoring functions. And search algorithms are based on systematic, sometimes it's molecular dynamics also, like people, some molecular docking programs are basically having short MD simulations during the confirmation search inside the binding pocket, and some local shape feature matching for getting the optimum binding orientation with respect to the interactions being made by the ligand and the receptor. That I will say, I will show some of the examples of the interaction in later slide, and sometime it is being done by genetic algorithm. Scoring functions are mainly based on the principle of force field, which is basically the mathematics or molecular mechanics equations of bonding between, basically non-covalent bonding, that is being estimated by the force field based methods. And empirical are nothing but the information by the experimental data. And the knowledge based is statistical methods are being used for the modeling of the scoring functions or estimating the binding affinities. And after these two steps done, you assess the docking and then find the docking accuracy, enrichment factor based on the available experimental data, and then predict the new compound structure, which can be active, more active than the compound being studied in this docking study, for example. So this is the real principle behind any molecular docking program. So structure-based drug design is basically the method which actually covers the structure of the ligand and designing the structure of the ligand based on the structure of the enzyme where it is going to bind. So you need to have knowledge of the 3D structure of protein, target protein for this particular case. And molecular docking is the best method in the structure-based studies. So same, showing you the principle how actually a ligand and protein or receptor are going to bind. It's a very simple lock and key kind of model. And protein ligand docking, and you use this for the purpose of designing of new ligand. And docking goal is to find a set of ligand which could bind with the receptors. And that set of ligand could never, maybe have never been tested against this particular drug target. So that's the real essence of having advantage of molecular docking methods or programs. And you check the validity of docking by redocking the co-crystal ligand inside the binding pocket again. If you get the RMSD value lower than 1, that is very, very valid or accurate docking pose coming up from the molecular docking program. So that's how you actually establish the validity of the docking program. So how and why this method is actually becoming so popular? Because as the time passing, you are getting so many information about the drug targets. And for the drug targets, we have found like about more than 45% drug targets are mostly the receptors from the cell, from the human cells or prokaryotic cells, whatever. But they are mostly the receptors, and 28% are around enzymes, and they both are protein in nature actually. So that gives a real advantage, or getting this information will lead definitely to find some methods to model this interaction between the protein and the ligand actually. So that's the real, I would say, basis of using my computational method for the protein design only. Most of the time you found like, docking is being done by having a protein with a ligand, So, but having said that, there are many other drug targets as well. For example, ion channels and transporters and others. And protein data bank is a central repository where actually every crystallized protein structure is being deposited with its coordinates, 3D coordinates, and you can see how this protein data bank evolved over the year. So in 2021, right, this year, we have about 200,000 protein structures solved crystallographically, and they are deposited in the protein data bank. This is a big resource to start such kind of molecular marketing-based research work in any way. It's freely accessible and available for anyone to be used for the purpose of drug discovery-based research. So for doing such kind of study, for example, structure-based study, you need to have a target structure, and that target structure need to have certain kind of characteristics which is required in order to start a reasonable molecular docking experiment, I would say. So for example, this kind of characteristic I have got from the literature, and this is found like most of the time the target should have a deep or enclosed binding site. It should fit at least ligands of a size of about 300 to 600 Dalton. So that characterize it as a good drug target to be studied by using computational tools. And it should have a mix of hydrophilic as well as hydrophobic characteristics. And for the docking purpose, the receptors or target structure are rigid. But if you talk about MD simulations, for which you need to have, you actually allow protein to be flexible. So what kind of chemical interactions a ligand is being made with the receptors are tabulated here. For example, hydrogen bonds, ionic interactions, metal ion complex, hydrophobic interactions, and some cation pi interaction and pi stacking is also considered as hydrophobic interactions. And these are the interactions which is being estimated during the by the scoring function of any docking program actually. So what kind of docking program are available and what kind of docking program are popular? Very much popular are represented here by highlighting in red color, rectangular, and some others are here. And there are many, but most popular are actually Autodoc, Ndoc, Flex, and Thread, Glide, Gold, and Surflex. And as I said, next step of any docking program is to score and estimate the binding energy. So these are the scoring functions which are based on different kind of principles, and popular scoring functions are also tabulated in this slide. And many famous scoring functions are highlighted by the red arrows. And another structure-based method technique is molecular dynamic simulation. What it does, it actually covers the limitation of the docking program, where actually the ligand is flexible, but the receptor was not. Here in molecular dynamic simulation, the system is whole flexible. So it gives you more realistic picture towards the binding interaction between the protein and the ligand, considering solvent factors, solvent interactions as well. So it allows you to find more accurate binding affinity of certain kind of ligand which you have never been tested before on that particular drug target. So it helps in bringing new drug molecules. This is actually a static diagram, but I think it is, I actually made, I got it from the website where actually they were moving. So it does not move here, it's fine. Okay, so you can just note the reference of this. So these are these molecular dynamic simulation images coming up from a study back in 2002, it was reported there. Okay, so virtual screening, what is that actually? So virtual screening is nothing but it is actually using multiple thousands of compound library in a method called structure-based methods or ligand-based method, where actually in a structure-based method you perform docking of maybe theoretically you can perform millions of compounds molecular docking and then score them. And then after estimating the scoring, after estimating the scoring is basically estimating the binding energies or the interaction energies between the ligand and the protein receptor, so that you can shortlist or you can rank like highly theoretically stable compound inside the binding site. So these are the things you are performed in virtual screening. So sorry, where's that? So okay, so a typical workflow for virtual screening is basically you start with a database of compounds. Literally, it has multiple thousands or multi-billion compounds library, as Dr. Saeed rightly mentioned last day about the Zinc database, which has millions of compound library, and they are not only available theoretically, but they are also available to be purchased. And you can, after performing your computational method, what you can do, you can buy them and then check them. So how well this virtual screening perform in reality, I will share some data later on in the slide, so you can believe or you can like use this for the purpose of drug discovery research. So you start with a database of having multi-thousand of compounds, and parallelly you actually prepare your target, and in the target preparation you perform hydrogen bond relaxations and other things, you know, ionization correction, tautomeric corrections, you know, and similarly with the ligands. And then you perform, if it is a structure-based drug discovery method. So, okay, so for the ligand library, I would like to say like they should be following the Lipinski rule of 5, which are mentioned here. So, and you perform molecular docking because it's a structure-based methods, and you come up with the, for example, you start with millions of compounds, you come up with thousands of compounds using this first step of filtering out the compound by using molecular docking, and then you perform scoring, and then you further short down compounds, and then you can use some top ranking compounds and purchase them and then really check them. So in a way you actually are reducing the cost and labor associated with your experimental methods in your research. So sorry. So there are some examples from literature are here mentioning here. So some different examples which actually people have found potent inhibitors against certain drug targets using these methods of structure-based virtual screening. And if I can get a literature example from the literature regarding the comparison with the high throughput screening, which is the real-time experimental assay of certain number of compounds. So it was found like the hit rate of an experiment, once it was actually pre-filtered by a docking program, is dramatically improved. So that gives you the real essence, like, you know, you are more focused using structure-based virtual screening methods. You are actually shortlisting some focused library of compound which are basically playing a role in the binding of the ligands, you know. So instead of going to the random searching of the compound, I would say, because technology is now advanced enough to actually can be used for the purpose of pre-filtering of the compound before actually really purchasing or synthesizing the compounds. Some of the examples of virtual screening from the literature where actually people have found some novel and unique drug candidates from the pharmacophore-based virtual screening. these are the mentioned in this slide, and the recent examples are here. So so far, I have been able, I would say, I was discussing about the topics of the tools required for early phase of drug discovery research. So from now, I would like to share one of my work which I have done using, which is a real example of an application of virtual screening on finding drug candidates against tuberculosis. So this is our own work, actually, so I would like to present here. And so TB is very famous and notorious disease, actually. And the problem is more, worst because the causative agent of TB is Mycobacterium tuberculosis, which actually got resistant to frontline antibiotics. So this is the major problem. And so drug-resistant TB is very common, and even in Pakistan, we are finding MDR patients and very few XDR patients. But in African countries, XDR, extreme drug-resistant TB, is a big problem. And another thing is that in the first world country, for example, in USA, major HIV patients are most of the time died by the TB actually because of the immunocompromised state of the patient. So this is. Actually, it's relatively old, so I should not be discussing. It's not the latest. So in Pakistan, it's still a problem of tuberculosis. TB is a big challenge, and we are still having many patients coming up. Although the treatment is available, but treatment is quite long. So there is always a room to find new drug candidates so that you can be treating the TB in more efficient manner with less period of drug therapy. So how actually MTB is unique? Mycobacterium tuberculosis is unique from other bacterial diseases because it has a very thick layer of cell wall, and that is comprised of different kind of lipid bilayers actually. So this is here. Due to that, drug needs to pass this big barrier of actually to treat the TB patient. The microorganism which is inside the body, the drug need to pass this barrier. That is a big challenge. And that's why TB has most of the drug targets are basically coming up for the TB are from the cell wall instead of cytoplasmic structures, because most of the time drug does not pass it actually. So this is the real, key of finding new drugs against MTB is basically targeting this cell wall. So there are many drug targets available against TB. What we have been actually, which we have picked up is basically pantothenate synthetase. That is why actually we have picked up, and because there are not much work was done against TB pantothenate synthetase, first of all. Second, the pathway in which pantothenate synthetase is involved in the Mycobacterium tuberculosis is absent in the human host. So that's why it's an ideal drug target. And no potent inhibitors of pantothenate synthetase has been reported. Still, actually, I would say there are not so many. So, and one more thing is basically it was found this pantothenate synthetase enzyme was found active during the dormant phase of TB. Even then, like TB, TB is famous for like having like many people are carrier of TB and they don't know. Sometime later in their life, they become immunocompromised and TB attacks on them. And during that period when TB is dormant, is basically still this enzyme was active. So if you actually are designing new inhibitors against this enzyme, you are basically targeting not only the active TB, but also the dormant state of TB. And that will lead to ultimately the complete cure of the disease from the patient. So at that time when we started this work, there were many PDB. There were not so many PDB structures were available against PDB, but against pantothenate synthetase in PDB. So there are quite like about 8 structures were available. They are reported here with the resolution not so. Actually, I would say 2 and beyond 2 is not considered as good resolution. It should be lower than that. And there were some missing residues as well. So simple overlapping resulting in finding some co-crystal water molecules, which are very, very important in terms of having the binding characteristic of the receptor structure. So these water molecules were also labeled here. And this is one of the images showing water molecules mediating interactions with the ligand as well as the receptor amino acids. And this is different views of the same binding pocket. And setting up two kind of receptor geometry. One is from 2A88, which is basically the apo structure of the receptor, and the 1 and 2I is the structure when it was bound with the ligand. It is having a co-crystal ligand. And then with different water occupancy, we have set up 8 possible receptor geometry, and we used commercially available database of from Novacore, LateQuest, Maybridge, and Compound, and ChemDiff actually. And altogether, it was 0.7 million compound structures. And what we were known at that time, there were about two actually inhibitors were reported from NIH, and these are here. And they were having quite an appreciable activity against this enzyme. And one of the reaction intermediate was being used as a reference for the purpose of, because it has the highest binding affinity towards the receptor. So these 3 were used as a known hits. And the docking protocol was set up. And these are the docking accuracy. And the real-time experiments were before performing the 0.7 million compounds screening. We have set up our docking protocol on 10,000 compounds only, so that we can set up our protocol and then run the large screening library later on after setting up and optimizing the protocol. So with that, we found about like the best performing receptor geometry was actually it's mentioned here. It's if you see, like, these are the ranking. This blue, green, and red are the known active compounds ranking. And if you see, like, shape gauge is the single scoring function which is giving in every receptor geometry almost pattern ranking of the known inhibitors. But this shape gauge is famous for basically is that if you see the structure is equation of this shape gauge scoring function, it is mostly based on the shape. So it does not really help in understanding the real chemical nature of the ligand and the receptor geometry. So we can use it, but not as actually, I would say, with accuracy. So if you see OE-chem score, which is more chemical oriented scoring function, and here if you see the receptor number 5, receptor number 7, they both are performed very well. So and giving the highest binding, highest rank of the known inhibitors, known compounds. So that lead to selection of either of these 5 or 7 receptors with the OE-chem scoring function. Okay, so consent scoring was also performed because the time is limiting me, so I'm not going to complete details, but this gives you further confidence of the setting protocol. And this is the, I would say, the scheme of this work, where these 10,000 compounds were used for the optimization of docking protocol, and later on we run this protocol on 0.7 million compound, and we got about 10,000 shortlisted compounds, which were high rank, top rank compounds from that. And then we performed short end dissimulation on these 10,000 compounds with single point energy minimization. And then we found, we estimated the MNPBSA calculations and find binding energies, and resulting in further shortlisting of about 217 compounds. And once these 217 compounds were experimentally checked, because we purchased those compounds from different vendors, and we ended up having about actually we found 5 or actually six experimentally proved compounds, in which 2 were found as a mixed inhibition with the other coupled enzyme acids. So we chose only the selective, which are 3D. These are the 3 ones. And okay, one may ask me, like, the question, like, the hit rate is very, very low compared to, if you talk about, like, we have started with 0.7 million compound. We ended up only 3 or 5 experimentally proved compounds. But still, the advantage of this exercise is that, like, with this, if you see these 3 compounds which were found in giving good activity against that target is basically they are having sulfur in their structure, and that helps us to make an SAR of these compounds and focus our future research on these particular, I would say, pharmacophore. So this kind of advantage you will get from, and similarly, we also have performed some kind of experimental medicinal chemistry efforts or synthesizing new analog of 2 hits, and from there we found few other inhibitors by considering the structural requirement. So I still have 10 minutes. Right? Less than 10 minutes. Okay, so so far I have been able to convey my message of regarding structure-based methods of finding new drug candidates. From next few slides, I would be talking about a reverse, I would say, methodology where we are trying to find, and we are also working in this area, and we are trying to find, instead of finding new drug candidates, we try to find new drug targets in the drug-resistant bacteria. So antibiotic resistance is a big problem. You know, many bacteria are now resistant to the frontline drugs, and they are actually not really getting treated by the drugs anymore. So I don't know if I want to go further detail. I would like to focus on my work actually, otherwise it will take much of the time. So this slide, I will focus on that. So why you actually want to focus on bringing new drug targets instead of new drug candidates? Because, you know, bacteria is already resistant to the already administered drug. So that means that bacteria has certain kind of mechanism, molecular pathways, which actually digesting that particular drug. So why not identifying new proteins in the same bacteria? Maybe they are using new pathways, so there are new set of proteins which are actually playing crucial roles for the survival of this bacteria. So that's why it's important to find new drug targets in order to combat the resistance actually. So we are focusing on finding and the normal mechanism how actually we found it. You see, many bacteria has many genes for which we don't know the function yet. So we try to find the functions by using bioinformatics-based methods, and if those unknown genes are coding such proteins which are hub proteins, like having big network of proteins around them, then it means they could be potentially acting as a new drug target. So that's the idea, and for which we are using bioinformatics-based methods, which involve this analysis. So one of the example I can show here, we have used this same method of finding new drug targets against the drug-resistant Pseudomonas aeruginosa, where actually we were able to find 68 first characterized unknown proteins into the known functions, and then out of which we were able to find eight new drug targets, which we have reported in this paper in 2018. And if someone interested can go and read it. And with that, I would like to conclude my presentation and summarize what I have been telling you so far. So computational method in MedChem represent the promising effect on lead discovery. Yes, there are lots of literature example, and our own work is also explaining this. And the combination of chemical information with virtual screening will play an important role in drug discovery, particularly in post-genomic era, where actually you are getting new genomic information every now and then. You are getting new information, and computational methods lead to much higher reach. And in addition, for computation methods, it is needless to collect and assay a great number of purified compound samples, and which dramatically saves drug discovery time and cost. And however, having said that, it has to be emphasized that computational methods are not the replacement of the actual experiment screening. As a matter of fact, these two methods are highly complementary. And some of the challenges still exist, like huge increase in the information of in the volume of information. Every day, every hour, you are getting new data coming up for the purpose of biology research. So biology is more like a data science nowadays, and for which we still have immature technology. We do not have technology enough to, I would say, solve the hidden information, to get the hidden information of this data. And there are lots of fuzzy science are still, like, for which we don't know how drug actually, how disease progress in the human body. So there are lots of challenges that still are there. So field direction, pharmaceutical history indicated that natural products provided a large number of drugs to the market. But for the currently used drug targets, available natural products have not been tested completely. So computational methods can help in them, like you can use computational methods to check them in computational methods, and then moving on further for experimental methods. And this is an example of first clinically approved drug from molecular modeling method, reported in this journal, and it is working as an active against HIV. And with that, I would like to acknowledge funding agencies which have been kind enough to provide us funding for the research, and my home institution, Penjwani Center, where I worked, and some universities where I have done my postdocs, UNSW, Germany, Heidelberg, actually. With that, thank you so very much, and I'm open. If you have any question, I can be happy to answer. Thank you so much. Hazima, are you there?
Yes, thank you very much, Dr. Riaz. It was a very nice presentation and interesting at the same time. With this, I think we have some questions in the chat box. Let me read them out for you. So first of all, Mr. Syed Lal Badsha is asking, have you performed any covalent docking?
I have never done myself covalent. I have read many articles regarding this covalent docking, but you know, covalent docking is possible and people do it, you know. But the problem happens once you rank it using scoring functions, you know. So there is no such scoring function which can, you know, find some appreciable accuracy to model the covalent interaction between the ligand and the receptor. So there are methods available. People do it. I have not done myself.
Right. So there is also another question by Dr. Fakhruddin. He's saying, number one, excellent talk. And secondly, he's asking how to identify unique drug targets, delivery targets.
Okay, so if I understand your question correctly, you are talking about how I identify unique drug targets. So unique drug targets are, if I go back to my presentation, if I think I can share again. Okay, so let me tell you without that. So what we do, you know, we start with complete proteome set of protein from a single pathogen, which is available, like thousands of pathogenic data is available from UniProt. And we use these complete set of protein, which is being expressed at one time by the pathogen. And then we perform search tools. We use search tools. For example, BLAST is very famous for that. But for that, you must need to use standalone BLAST, actually. You cannot use online BLAST, which is actually taking so much of the time. So I use standalone BLAST, and then I automate it by using shell scripting, and then I get within 6 hours or 7 hours, I can get complete similarity index of one pathogen proteome data set with the human set of proteins. So in this way, I would be able to find at least first question I could answer, like which protein in pathogen is unique and not found in human host. So it gives you that answer. And then from that, you move on to other kind of steps. So first step is that, and that is the crucial step of finding unique drug target.
Thank you very much, Dr. Riaz. I think we can take one more question. We have 2 minutes. Mr. Mehdi Hassan is asking how the output data from virtual screen is validated.
Okay, so as you remember, I have talked about known inhibitors, So one of the, I would say, limitation of any docking-based program is basically you should have at least few known inhibitors or known ligands, which are actually, like they are really binding with that. So with that, you can use them as for the purpose of finding the enrichment factor. For example, if you have thousands of unknown compounds that have never been tested against that particular drug target, but you have 10 compound for which you have activity data of that particular drug target. So you can use that 10 compounds data set out of these thousands of compounds and see how well they are being ranked by this virtual screening method. So if they are getting ranked well with appreciable accuracy, you can estimate by using. There are lots of methods, by the way. There are lots of statistical methods to do that. For example, enrichment factor, for example, ROC curves, If you get better statistics regarding these known inhibitors, then you say this method is valid. If there is low ranking of these known inhibitors, then it is low. I would say it's invalid, actually. That's how we do.
Thank you, Dr. Riaz. I think we have another question. I think this should be a brief one. Give me a minute, please. So the question was that are the databases for virtual screening free?
Many of the database for virtual screening is free. Like, for example, Zinc database is freely available, and some commercial companies also provide. I use commercial database, by the way. So for which you need to talk to the company and then ask them to provide you the structures catalog of these compounds, which are available in structure description format file that is freely available. This is nothing harm for any company to share it.
Thank you very much, Dr. Riaz. And I think we can take one more question. What if a pathogen target has a homology to humans, but still is an important enzyme for that pathogen? Can it be considered a target?
Yeah, that's a very, very good question. I really wanted to ask, really wanted to answer this question, actually, so I read this question. So, okay, but so the answer of this question is that, yes, you are right. There are many proteins which are sharing the homology with the human host also. But if it is having like a very high similarity, very, very high similarity, like more than 80%, 90%. So that means if you are targeting that particular protein for the purpose of drug acting on the pathogen, then you are harming the human metabolic pathway as well. That leads to side effects, So the ideal is to get those particular enzyme or proteins from the pathogen which are having no homology with that. But if you still found an enzyme which is actually having similarity or homology with a human host, then you try to find those ones actually which are very, very low in similarity. And if still they have similarity, for example, if I talk about pentothenate synthetase, which I have targeted, so there is no pathway available for this kind of vitamin B5 metabolism in the human, but it was having about 40% similarity with one of the protein found in human host. So if your targeting protein is having about 40 percent similarity, you are safe, still safe to target, and you need to see the binding site difference of that particular protein of homology with human and a pathogen. Although if they are more than 60%, 70% similar, but they have very different binding affinity, binding activity, binding cavity actually, binding interaction enzyme, binding interaction amino acids, then still you can target this as like a drug target. But if they are sharing the active site also, then you cannot use it as a drug target. So this is the rule of thumb.
Thank you very much, Dr. Riaz. With this, again, thank you very much for taking out your valuable time and for delivering this important lecture. With this, we will move on to our next speaker, Professor Dr. Jamshed Iqbal from Comsets University. And with this, the title of his talk today is Discovery and Pharmacological Evaluation of a Novel Series of Adventile Thiourea as G protein coupled receptor antagonists. And to formally introduce and welcome him, I would request Dr. Hina Siddiqui to introduce and welcome our guest speaker.
Thank you so much, Khazima, and it's such an honor to introduce a person of stature like Professor Dr. Jamshed Iqbal Sahib. It's greatly, truly an honor for me. Professor Dr. Jamshed Iqbal Sahib is Chairman, Department of Pharmacy and Head of the Center for Advanced Drug Research at Comsets University, Islamabad, Ahmedabad campus since 2007. Professor Iqbal has completed his early education from Pakistan, while he got his master's and PhD degree from University of it's very difficult name, university in Glasgow, UK, and University of Bonn, Germany, respectively. After doing 2 postdocs from Germany and China, he opted to serve Pakistan and joined Department of Pharmacy, Comsets University, Islamabad, Ahmedabad campus. He has made notable contributions in pharmaceutical chemical sciences. Professor Iqbal has more than 270 publications with cumulative impact factor exceeding 850 in medicinal chemistry, enzyme kinetics, natural product chemistry, drug discovery and metabolism, molecular modeling, and molecular toxicology. In addition to research publication, he has 2 international patents. He has supervised over 50 PhD and master scholars and earned over 250 million PKR research funding from higher education, Pakistan, comstech-oic, OPCW, DART, Germany, and other sources. His research, his scientific and capacity building contribution have been recognized by prestigious national and international awards and honor and fellowship of several academies of science and technologies. According to the Research Productivity Award, RPA list issued by Pakistan Council for Science and Technology, Professor Iqbal was awarded grade A, category A awards. And he was also awarded Pakistan Academy of Sciences Gold Medal by Pakistan Academy of Sciences. And in 2021, Ministry of Science and Technology and the Ministry of Foreign Affairs Government of Pakistan has appointed him as the member of the task force for science diplomacy. In addition to his research activity, Professor Iqbal has served the Comsets University Islamabad as a member of Board of Advanced Studies and Research, member of Academic Council, Departmental Advisory Committee, and convener of Research Productivity Award Committee. Professor Iqbal has served as editorial board member and reviewer of several international journals with high repute. With this introduction, I request a very senior professor, Professor Dr. Jamshed Iqbal Sahib, for his lecture. Sir, over to you.
Yeah.
Now you listen to me, I think. You hear me?
Yes, sir.
Yes, sir.
Okay, Assalamu alaikum and good afternoon. Thank you very much for a very kind introduction. So the title of the presentation, Discovery and Pharmacological Evaluation of a Novel Series of Adventile Thiourea as G protein couple receptor antagonist. Actually, when I prepared this presentation, I was thinking something other, but then we planned to make a patent of this work. So I will deviate on the compounds to some extent. So there, according to the Nature Reviews drug discovery, there were several biochemical classes of drug targets. Among them, the first and the largest is the receptors, 45%. Among the receptors, the glycoprotein couple receptors, they are the 25%. Enzymes is the second largest class of drug targets, 28%. And similarly, nuclear receptors, ion channels, DNA unknown, and some hormone and factors. So what is the function of receptors? P2 receptors have been found to be expressed in virtually all cells and tissue types. Their function is to mediate an array of biological functions. It includes platelet aggregation, smooth muscle cell proliferation, and immune regulation. And they also has several, many functions in the body. The purinergic receptors, they have been classified into two main groups. Although there are three classes, but the two are the main prominent classes. The P1 receptors and P2 receptors. P1 receptors, these are called adenosine receptors because adenosine is the natural agonist of these receptors. So they are A1, A2A, A2B, and A3 receptors. These are called G protein couple receptors. The P2 receptors, they are again divided into two classes, P2X and P2Y. P2X1 to P2X7, P2Y1 to P2Y14. And the P2X receptors, these are called ligand-gated cation channels, and P2Y receptors, they are called G protein couple receptors because they are coupled with some specific protein, so that's why they are called G protein couple receptors. Among the P2Y receptors, they are further divided into two subgroups, P2Y1, P2Y2, P2Y4, P2Y6, and P2Y11. They are similar in their structure and in their function. They are similar, that's why they are grouped in one class. Similarly, P2Y12-like receptors, these are called GI receptors, P2Y12, P2Y13, P2Y14. So these are the three, they are related to each other. So in P2X receptors, there are some homologous receptors and then there are some heterologous receptors. The properties of P2Y receptors, their tissue distribution, their agonist, and their G protein in which they are associated for their function. P2Y1 receptor, they are widely distributed, including the platelets, central nervous system, heart, skeletal muscles, GI tract. So their natural agonist is the adenosine diphosphate. And then P2Y2 subtype receptors, it is widely distributed, including lungs, heart, skeletal muscle, kidney, brain. Similarly, P2Y4, they are distributed in placenta, lungs, vascular smooth muscle, and their agonist is UTP. So they are mainly ADP and ATP, UTP. These are their natural agonist. So P2Y6, P2Y11, similarly P2Y12, it is mainly expressed in the platelets. Their high expression is in the platelets and in the brain. And their natural agonist is the ADP. Similarly, P2Y13, P2Y14. Sorry. So if we see the structure of the G protein couple receptors, there are 7 transmembrane domains. And with the C terminal in the cytosolic side and N terminal is the extracellular side. And this is their mechanism when the ligand is attached with the receptor, then it initiates the the functions, and then it will release the calcium. So this high amount of calcium is associated with different pathological conditions like digestive cancer, pain and inflammation, Alzheimer's disease, cardiac fibrosis, platelet aggregation. Actually, at the cell surface, we have receptors as well as enzymes. So this is the main problem, that sometimes the agonist is also the substrate of the receptor. So ATP is released from the cells under high stress or with some cellular injury. It is released outside in the extracellular space. And ATP is the substrate of the antipedases, nucleotide triphosphate diphosphohydrolases enzymes. And ATP is agonist of the P2X receptors. These are the ligand-gated ion channel receptors. These are the trimeric receptors. So when ATP is attached with the P2X receptor, it will release the calcium, and then it starts the diverse cellular responses. And ATP is the substrate of the antipedases, which convert ATP to ADP, which is the agonist of the P2Y receptors. And then ATP is also the substrate of subclass of antipedases, and they convert the adenosine diphosphate to adenosine monophosphate, which is not the agonist of any receptor. However, once actin 5 nucleotidase convert adenosine 5' monophosphate to adenosine, it acts as the agonist of the P1 receptor. So once we have the good agonist, it is possible that it is degraded by the enzymes. So once it is degraded by the enzyme, it can be converted into the agonist of the other receptors. So then we feel that we suffer the problem with reference to the selectivity. So most in ideal cases, the agonist should not be hydrolyzed by the enzymes, and it should be selective for the receptor. So this is the main problem with the receptor and the enzyme, the selectivity, because these enzymes and the receptors, they are very much closely related to each other. So this is the mechanism of activation of purinergic receptors. ATP is converted to ADP with the release of inorganic phosphate, and this it acts as agonist of the P2Y receptors, and it opens the calcium channel, and it releases the calcium inside the cells, and this start different pathological diseases. So the P1 receptors, they are 7 transmembrane receptors with the disulfide bond, and this is the binding site of the agonist, and the nitrogen terminal is at the extracellular site, and carbon is in the intracellular site. So this is the P2X family receptors with the 2 transmembrane domains. Both nitrogen and the carbon, both are inside the cytosolic region. However, in case of P2Y receptors, we see several color dots, which shows their specific amino acids which are conserved, and some amino acids which are binding by the agonist. So the major challenge to develop the different selective and the potent antagonist of the P2X and P2Y receptor is the selectivity. There is a lack of selective ligands, agonist and antagonist, for many of the P2Y receptors. This is the issue at the moment, because there is a very closely related subtype of receptors, and there is no selective agonist. So mostly there is non-selective agonist or antagonist. And if we have nucleotides as a agonist, they are fastly degraded by the nucleotides enzymes. So if we have the ATP derivatives of act as an agonist, it can be hydrolyzed to ADP and further to AMP by the enzymes. So we have a problem with the selectivity of the specific agonist. Similarly, cells can release nucleotides when they are being stressed by physical movement, stirring, shaking, subsequent to change of the incubation medium, or by changes of the pH value. Once the cells are in the stress, they release the nucleotides. Once they are released, then they undergo different mechanisms to degrade into ATP, AMP, and adenosine. Molecular modeling and high throughput screening approaches we designed to systematically analyze the structure activity relationship of the compounds, and then we further modified them and optimized to get the potent compounds. The objective was to focus on the non-nucleotide activated P2Y receptor subtypes, P2Y2, P2Y4, P2Y6 receptors, and ADP activated P2Y12 receptor and P2X subtypes. So the goal was to find new and subtype selective agonist for P2Y and P2X receptors, and to study the structure activity relationship of agonist at those receptor subtypes. And we also develop the homology models, and we also do some virtual screening, and then we find the leads, and then we design the agonist or the antagonist, and then we test them in our system. So this is the one of the drug which is in the market. It is a selective P2Y12 antagonist. It inhibits ATP-induced platelet aggregation and displays antithrombotic activity. It is used to reduce the risk of heart attack. So clopidogrel is one of the drug. It is a selective P2Y12 antagonist, but it has some drawbacks, like it is a prodrug and it is metabolized by the enzyme cytochrome P450, and 85% is not available. Only 15% act as an antagonist on the P2Y12 receptor. There is another drug which is in the market. It is a selective P2Y12 antagonist. It is a ticagrelor. It is also a prodrug, and it is metabolized by the P450 to convert into the real drug. So this is another example where we can use the P2Y6 receptor antagonist as a drug. P2Y6 receptor activation promotes inflammation and tissue remodeling in pulmonary fibrosis, and also treating inflammatory bowel disease by blocking P2Y6 receptors. So antagonist of the P2Y6 receptor can act as a drug for the treatment of inflammatory bowel disease. So normally we express the different receptors on the astrocytoma cells, 1, 3, 2, 1, and 1 cells. So this is the procedure how we express the specific receptor on the cell lines. These are the reported antagonists of the P2Y receptors, like PPTU. It is allosteric antagonist, and PPADs also, but these are non-selective. Reactive Blue2 is also very good antagonist, but again it is non-selective. Soramine is also non-selective antagonist. So mainly we are suffering from the selectivity issue. So we selected the indomethacin, which is a non-steroidal anti-inflammatory drug, commonly used to reduce fever, pain, stiffness, and swelling from inflammation, because we see one of the derivative of very closely related compound, which is the P2Y12 antagonist. So this is indole carboxamide compound, and then we synthesize this derivative, different derivatives which act as the antagonist of the P2Y receptors. So most of the compounds, they were selective at the P2Y1 receptor. So these are the dose response curves, which shows the selectivity potency of the compounds. So soramine we use as a non-selective antagonist, and these are the other our synthesized compounds, which shows the antagonistic activity against the P2Y1 receptor. Unfortunately, we did not have all human subtype receptors. This is the P2Y1 from the Turkey. And this is the data with the human P2Y2 receptor, in which we use the reactive GLUT2 as an antagonist, and this the dose response curve of the different compounds. Again, this is the human P2Y4 receptor with the different dose response of the different antagonists of the indomethacin derivatives. So we also performed the molecular docking study to confirm the binding interaction of our compounds in the active site of the receptor. So this is another compound 4H. It was the most potent compound and selective compound at P2Y1 receptor. So again, this is the Turkey P2Y1. We did not have the human P2Y1 receptor. So maybe it can have some selective issue as compared to the human receptor. And this is the interaction of the 2 and 3D dimensional interaction pose of Turkey P2Y1 in antagonist for I compound. So this is the data of all the compounds using the different thiourea derivative of indomethacin. They are substituted at this position. So we have some compounds, they show some selectivity, and there are some compounds like in P2Y1, this was less active, only 40% activity. And similarly at P2Y4, it was 44%. So we did not get the IC50 value. So this is at human P2Y2, it is the, we get the IC50 value. So we compare the data of different P2Y receptors, subtypes, using the different thiourea derivatives of the indomethacin. So this is the compound 4H, which was the most selective and most potent in antagonist of the P2Y1 receptor with the 360 nanomolar potency. And it is less potent at the other receptors. Like P2Y2, it is only 5.39 micromole, and P2Y4 and P2Y6, it was only some 43% or 48%. So we use the soramine in some cases as the standard antagonist, and reactive GLUT2 was also used in some cases as a standard antagonist of the receptors. So we also synthesized some compounds, and we tested on the P2X receptors, which are the ligand-gated ion channel receptors. So these are the trimeric channel. And when the ATP binds with the channel, it opens the calcium, and calcium is released inside the cell. So we have the sulfur disulfide bonds, and it is a very big loop in which the ATP agonist can bind at the hydrophobic residues. So these are the compounds which are the antagonist of the P2X3 receptors. So this is the mark. They just their application is accepted by the FDA on 1st March 2021. They, I think, successfully passed the clinical phase 3 trial. So this is for the treatment of chronic pain cough. So these are the highly potential target for the for specific diseases. So in case of market analysis and insights, it is the forecast that in 2027 there will be 112.7 million US dollar budget for revenue will be for this P2X7 receptor. This is the most highly studied receptor in P2X family. So it has a role in cancer and inflammation and chronic pain and neuropathic pain. So these are the P2X7 receptor antagonist market segmentation by application, inflammatory diseases, then central nervous system, immune system, neuropathic pain, and cancer. So there are several drugs which are in the clinical stage in case of P2X7 antagonist, but none of them is approved yet. So these are the pharmaceutical companies. They are the top player in case of P2X7 receptor antagonist, like GlaxoSmithKline, AstraZeneca, Johnson, and so on. So in case of activation of the P2X receptor, it starts the cellular death and inflammation by renal and vascular dysfunction. It produces the hypertension, and then atherosclerosis development. It causes the stroke and cardiac fibrosis and dysfunction. It results the heart failure. So there's the several conditions in which P2X7 antagonist can be used as a drug. Also P2X3, they have a role in diabetes, and we also studied in our lab the role of P2X3 in diabetes using the isolates of Langerhans. So these are the some antagonists of the P2X receptors. Normally we search the literature and we see the most potent compounds, and then we try to mimic or try to find the suitable derivatives which can act as the potent P2X subtype receptor antagonist. So these are the P2X antagonists which are in the clinical trials against inflammation, pain, cancer, and arthritis. So these are the agonists, mainly agonists. They are like nucleotide derivatives or various, they are nucleotides, substituted nucleotides or nucleotide derivatives. They act as a agonist. So in case we have benzyl ATP as a agonist of the P2X receptor, and alpha beta methylene ATP and alpha gamma methylene ATP. So these are the agonists, and they are also stable against enzymes because their alpha beta methylene ATP is not degraded by the enzyme. Similarly, alpha gamma methylene ATP is not degraded by the enzyme. There are some examples of the non-selective antagonist for the P2X receptors in which the thiourea bond is present. So there are sulfonate and thiourea linkage which are responsible for the activity of the antagonist. So in case of soramine, which has the urea linkage and also sulfonates, it was studied as a agonist of different receptors, and it shows some moderate selectivity with respect to the different receptors. So reported urea derivative as P2X receptor antagonist. Actually, we were interested in the urea derivatives which can act as antagonist of the receptors. So therefore, we survey the literature with the urea and other moieties like admintyl and so on. There are already reported compounds against the different receptors which are selective and potent. These are some sulfonamide or sulfonate based compounds which are the agonist of the different receptors P2X, P2Y subtypes. So similarly, like we make the assays for the P2Y receptors, we also establish the functional assays, calcium mobilization assay for the P2X receptors. So this is the protocol in which how we make the activity assay of the receptors. So this is the basic principle. We have the Fura-2 dye, which is when passed through the membrane, and it is then by the esterases, this active form make the chelation with the calcium, and then we absorb the fluorescence. It is very selective and efficient assay. So we make the expression analysis of the P2X2, P2X4, and P2X7, P2X5 receptors using they were expressed in the human astrocytoma cells, and we analyze their real-time PCR and gel electrophoresis to verify their bands to verify their expression. So in the assay, first we use the cell lines, and then we use the ATP or the benzyl ATP as the natural agonist of the receptor, and then we see with the non-transfected cells, we did not see any signal. Then we have like P2X2, P2X4, P2X5, and P2X7 receptors. So we compare these all receptors, their expression analysis using their natural agonist. So these are the some P2X receptors agonist which we tested on the receptors. So these are the sulfonamide, third heteroaryl ethyl dienes. So they are the two series of the compounds which we tested on the P2X subtype receptors. So first of all, we tested the standard compounds like soramine, PPADs, and some other compounds against the different receptors, and we then established their dose response curve. So on the left side, we have the dose response curve of the ATP with the P2X2, P2X4, P2X5, and P2X7. P2X7, we use the benzyl ATP as the agonist. Similarly, for the soramine, we use the soramine as an antagonist, which is non-selective, and it is this dose response curve is for the P2X2, P2X4, P2X5, and P2X7. There are some P2X receptors which we could not study because their response time was too fast. So this is with the PPADs, pyridoxal phosphate azophenyl disulfonic acid. So these are PPADs against the for the three different receptors. So this is the data comparison of the different derivatives of both series with the P2X2, P2X4, and P2X5 and P2X7. So luckily at this time we managed to get the plasmids for the all human receptor subtypes. So here we can see there are some compounds, they are less potent at one receptor subtype, but potent at the other. So like 6B, it is highly potent at the P2X2, but it was less potent at P2X4 and P2X5. But again at P2X7, it was slightly less potent than the P2X2. So we got some selectivity in the derivatives. So these are the other derivatives of the series which we tested against different receptor subtypes. So this is the ethidium bromide diuptake assay for potent inhibitors of the human P2X7 receptor. So in one case we use the benzyl ATP without inhibitor, then benzyl ATP with 20 micromole, one antagonist, then benzyl ATP with 20 micromole of the second antagonist. So we also performed their docking studies to verify their binding mode inside the active site of the receptor. So there also we have problem with the docking studies because their crystal structures are not available. So mostly homology models are present, or maybe we select the crystal structure of one receptor and then we make the homology model based on that receptors to study the molecular docking study for our compounds. So this is the compound 7E, which we dot with the reference compounds. Similarly, there is another compound which we make the docking studies with the active side of the receptor. So once we finish with the receptors, we also studied these compounds as a inhibitor of the actonucleotidases, because enzymes and the receptors, they are present at the extracellular environment of the cells. So we have different enzyme subtypes, like we have nucleoside triphosphate diphosphohydrolases, antibidase. So these are the 8 enzyme antibidase 1, 2, 3, 4, and 8. 4 enzymes. Similarly, nucleotide pyrophosphate phosphodiesterase, we have NPPs 1, 2, and 3. And similarly, we have alkaline phosphatases and acto 5 nucleotidase, which product is adenosine, which is the agonist of the P1 receptor. So we synthesize the compounds and also we investigate on the different receptor, on the different enzymes, whether they are the inhibitor of the enzymes or whether they can be decomposed by the enzymes and they can be degraded by the enzyme and can result into the other agonist or the antagonist. So again, this is the structure of the different enzymes. They are present at the cell surface and mostly they use the ATP as a substrate. Some cases they prefer the ATP, in other case they prefer the ADP. And so ADP and the ATP, they are the agonist of the P2 receptors and also they are substrate of the enzymes. So at the one time it is an agonist and at the same time it is a substrate of the second enzyme. So it is very important to have the compound which is non-hydrolyzable by the enzymes, but it should be the selective and potent agonist of the receptors. So these are the different classes of the antipedases. Again, they mostly use the ATP, adenosine, ATP, adenosine triphosphate, and as the substrate is common, so there is a very high similarity against among the different antipedases subtypes like antipedase 1, antipedase 2, antipedase 3, antipedase 4, 5, and 6. So for these different enzymes, we also synthesize the compounds and we test their selectivity, and then we also test at the receptors as a agonist or the antagonist of the receptors. So like receptors, antipedases, they also have some pharmacological actions in the body, like they involve in several processes including vascular hemostasis and inflammation and similarly thrombosis, and they are involved in inflammatory responses. They are present sometime at the surface of the regulatory T cells. Some are highly expressed at the beta cells. So they have their role in the different diseases because of their presence in the different tissues in the body. So this is the structure of the nucleotide pyrophosphate phosphodiesterases. These are the mainly three enzymes. They are most important enzymes because they use the substrate at a lower micromolar Km value. The NPP, other NPPs, nucleotide pyrophosphate phosphodiesterases, they use the very high concentration of the substrate, so they are not a really good target for the drug development. So they are the different classes of the NPPs with a reference to their similarity with reference to their substrates. So this is our group. I'm thankful to the students who participate in the different projects and they work hard to achieve the results. And I'm very grateful to our international collaborators. Sometimes they provide the plasmids, sometimes we get the homology models, and sometimes we get the cell lines and so on. So we are very grateful to our international collaborators for their help. And finally, but not last but not least, we are thankful to the funding agency, Comstech University. They provide the research grants for the chemicals and the for scholarship for the students. And we have several projects from the HEC. And also we have a project from the OPCW organization for the prohibition of chemical weapons. And we have several projects from the German exchange program, DRD, and also from the World Academy of Sciences. And currently we also have project with the France, and we are very much thankful to our funding agencies for their provision of fund. Thank you very much for your patience, and if you have any questions.
Okay, Khazima, do we have some questions? So I think we can take the questions now. Kazima, are you around? Okay, in that case, sir, I can read some questions for you. For example, I have a question from Hassan. He's asking how to confirm that it was exactly the synthesized derivative that inhibited the P2Y2 receptor or other receptors as antagonist.
Yes, we perform the functional assays for each receptors. We express every receptor on the cell lines, and then we perform the functional assay like a calcium mobilization assay. It is very selective and specific. So we inserted only the specific gene for that receptor, and that is expressed, and we verify our expression using the real-time PCR and other techniques.
Okay, okay. I think rest is all the appreciation for this excellent talk, sir. And few people have requested for the yeah, okay. So many people have mentioned their greetings to you for sharing such an informative talk. Thank you so much, sir, for sparing time for this event, and we're looking forward to have this type of interaction in future as well. Thank you so much.
Thank you very much.
So, Kazima, are you around now? I think I have lost contact with Miss Khazima at the comstech. So with this, I think we reached to the final stage of our workshop come online meeting. And I think it was such an overwhelming experience to have lots of participants throughout the 2 sessions. We lost some of them, but still we maintain good strength of participants. And we had very exciting and interactive 2-day session. And Kazima, are you around? I saw hand raised.
Yes,
I was trying to contact you.
Yeah, I'm
Okay,
sorry, I
over to
was
you.
away from my computer. So
No problem.
I would also like to thank Dr. Jamshed as well for his valuable time, and I'm sure this his talk would have been excellent, owing to his vast experience in this field. And Dr. Hina, do we have questions in the chat box?
Yeah, there were a few I have asked, sir, and I think we need to now conclude the online meeting.
Okay, okay. Dr. Hina, I think we should start from you if you have something to say regarding this workshop, and if you think it has,
Right. So, yeah, as I was just mentioning that it was very nice to see that we maintained the good strength of participants throughout the 2-day session, and each speaker resource person have put lots of efforts, and they deliver whether the academic or scientific talk in such a nice manner. And certainly this will lead to lots of collaboration, because while we were having their lectures, I saw many people, scientists across different countries were interacting together, and they were willing to initiate collaboration. So I think this fulfilled the requirement, fulfilled the main objective of this event under the comstech in collaboration with ICCPS, that the the faculty member, the scientists across different countries, they need to interact and they need to create collaboration, and based on that the young scholars, young students will eventually learn. So this event initially was planned as a workshop, and we all understand workshop has a different format. It was initially planned as a hybrid mode, but because of the recent emergence of increased number of Delta variant cases, we just shifted this into this completely online version. So we are planning to have a workshop in drug designing, especially with the respect of medicinal chemistry. That will be like a real workshop, and not the scientific content, not the scientific or the research work will not be there, but largely we will discuss about like how the molecules are being designed, what are the tools available in medicinal chemistry, which we try to convey in this one as well, but we will try to have in the physical manner at the comstech, inshallah, and maybe hybrid as well, and we will have more of interactive session and some hands-on training as well. So this I would like to thank all the participants for their presence, their participation, and I wish all of you a very good luck for your future. Thank you so much. And I would like to thank Hazima also, because she has been very instrumental throughout the event, and all the coordination she was doing was excellent, wonderful. And on top of it, I would like to thank my dear teacher, Professor Dr. Mohammad Iqbal Chaudhary Sahib, for his kind support, encouragement in organization of this event. And also I cannot forget to thank all the resource persons. They were amazing. They spare their time despite of difficult time zones and busy schedule. So thank you so much, everyone.
Thank you very much, Dr. Hina. And I would also like to thank you for your, tireless contribution in this whole today event. And I would also like to thank our speakers who had delivered such an informative, exciting, as well as impressive presentation these 2 days. Last but not least, I would like to thank hugely to Professor Dr. Iqbal Chaudhary for being with us for all these 2 days and for being, supportive of this event. And with this, I would request Professor Dr. Iqbal for his concluding remarks for this event.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Hazima and Hina, thank you very much for concluding already. And I would have only responsibility of thanking you, both of you. Have done wonderful job of putting together an excellent program with very prominent speakers from 3 different continents. And I'm absolutely delighted that till the end, we have some 200 people stayed on, which actually reflect not only the quality of event, but also the organization of the event in a manner that it become a very holistic in nature. So it was a journey from historical background to discovery to design. Medicinal chemistry has emerged as an excellent tool, and everyone appreciated the fact that this field is contributing in human well-being at the face of all diseases and pandemics, and is going to remain a very important discipline for all of us to develop our indigenous capacity. What I would like to request as a follow-up, Hina already mentioned that we would have more of such event focusing on various aspects of medicinal chemistry. But to begin with, I would request resource persons to kindly mentor some of the participants. So you can send your request to Hazima and Hina if you would like to seek advice on your medicinal chemistry projects. And we would request our resource persons from different institutions to guide you in your work. Maybe this collaboration would eventually emerge into visits from one place to the other, which Comstech would be very happy to sponsor and facilitate. With this, I would like to thank all of you for your presence and would like to especially thank the speakers from China, from USA, from Turkey and Pakistan for their excellent presentation. I think it become a permanent record of excellent science and excellent presentations. Thank you very much. Thank you.
Thank you, sir.
Thank you very much. Dr. Hina, do you have anything to say?
No, it's okay.
Okay. Thank you very much again to all our speakers, as well as to our participants who stayed with us for all these 2 days. And with this, I would like you to know that we will be sending certificates to the participant who attended both the days of this event in a week's time, inshallah. And with this, thank you very much for joining us and hope to see you soon in some other event of the Comstech website, Comstech. And we would also like you to visit our Comstech website, Facebook page, and just for the update. And let's stay in touch. And thank you very much for joining us.
Thank you.
Khuda Hafiz.
Hafiz, thank you.